Muscular tissue. What are the tissues having properties of excitability and irritability? Muscle tissue and neurons and nervous tissue. What are the tissues having properties of excitability and irritability? Muscle tissue and neurons and nervous tissue. Why muscle is contractile? Answer. Due to the presence of myofilaments, especially thick and thin filaments, which are composed of contractile proteins, myosin, actin, troponin, and tropomyosin. What are the non-muscular contractile cells? Myoepithelial cells and myofibroblasts. So, myoepithelial cells and myofibroblasts. Classify muscular tissue. Classification of muscular tissue. Morphological classification. Striated muscle, skeletal muscle, cardiac muscle, non-striated muscle, smooth muscle. So classification of muscular tissue, morphological classification, striated muscle, skeletal muscle, cardiac muscle, and non-striated muscle, smooth muscle. So these are morphological classification. And then functional classification, voluntary muscle, skeletal muscle, involuntary muscle, cardiac muscle, smooth muscle. So voluntary muscle, skeletal muscle, and cardiac muscle, smooth muscle. General classification: skeletal muscle, cardiac muscle, smooth muscle. Attached to the skeleton, forms the wall of the heart, forms the wall of the viscera. Skeletal muscle characteristics: cells are elongated, cylindrical, multinucleated, nucleus is oval, located on the periphery, subsarcolemmal, striated due to alternate arrangement of actin and myosin filaments, permanent cell, usually under voluntary control, supplied by somatic nerve. Example: muscles of limbs and wall of diaphragm. So these are skeletal muscles. And then development develops from the myotome part of the somite. Paraxial mesoderm develops from the myotome part of the somite. Development from myotome part of somite. Functions: contractions for locomotion. Functions: maintenance of posture, support and protection of internal viscera, heart production, modeling of bone, maintenance or stability of joint, helps in expulsive acts, peripheral located nuclei, prominent transverse striations. Cardiac muscle characteristics: composed of elongated branched individual cells that lie parallel to each other, striated but less distinct, presence of intercalated discs at sites of end-to-end contract. Structures found only in cardiac muscle. Each cardiac muscle cell composed of one or two centrally located pale staining nuclei, supplied by automatic nerves. Contraction is involuntary, vigorous, and rhythmic. Example: myocardium and muscles of great vessels joining the heart. Development develops from splenopleuric layer of parietal plate mesoderm. Functions: pumping blood out of heart, contractions of pressure regulation, branched fiber central nuclei, intercalated disc, cross striations, spindle shaped muscle fibers, centrally placed single nuclei. Smooth muscle characteristics: consists of collections of fusiform, spindle-shaped cells that do not show striations. Each cell has a single nucleus located in the central of the cell's broadest part, supplied by autonomic nerves. Their contraction process is slow and not subject to voluntary control. Example: alimentary, respiratory, and urogenital tracts, main ducts of glands, blood vessels, lymphatics, dermis, ciliary body, iris, etc. Development develops from a splenopleuric layer of lateral plate mesoderm. Functions: movement of viscera, contraction for propulsion, Gives the histological difference between different types of muscle tissue. Traits: skeletal muscle, cardiac muscle, smooth muscle. Long and cylindrical. Long cylindrical cells which are joined end to end provides side branches. A small spindle or fusiform in shape. Branching of fibers: absent, present, absent. Intercalated disc: absent, present, absent. Branching of fiber: absent, present, absent. Intercalated disc: absent, present, absent. And nuclei on the skeletal muscle, there is multiple and peripherally placed, one or two centrally placed, single centrally placed. Cross striations: nucleus multiple and peripherally placed, one or two, single centrally placed. Cross striations: striated due to alternated arrangement of actin and myosin filaments, striated but less distinct. Non-striated due to the lack of alternate arrangement of actin and myosin filaments. Sarcolemma: definite sheath, thin sheath, very thin sheath. Sarcolemma. T tubules present, small, involved in triad formation. T tubules present in large, involved and dilate formation. Absent. Sarcoplasmic reticulum: well developed, less well developed, rudimentary. Sarcoplasmic reticulum. Control: voluntary, involuntary and involuntary. Key functions: movement of the body parts, pumping of the blood from heart and movement of viscera. Contractions: quick, forceful, vigorous and rhythmic. Slow. Development: paraxial mesoderm, splachnopleuric layer of the lateral plate mesoderm and splachnopleuric layer of the lateral plate mesoderm. Mitosis: absent, absent, present. Capacity for regeneration: limited, involved in sessile cells only. Very poor, good, involved in mitotic activity of muscle cells. Nerve supply: somatic, automatic and automatic. Muscles of distribution: leaves and wall of diaphragm. Myocardium and muscles of great vessels join the heart. Present in the wall of hollow viscera, for example, gastrointestinal, respiratory and immunogenital tract. Cell response: cell response to increased load. Hypertrophy: increase in fiber size. Hypertrophy and hyperplasia: increase in cell fiber number. So nucleus and nucleus. Muscle: smooth muscle, intercalated disc, striations, skeletal muscle fiber and striations. Nice to know. Development of muscle: all muscles of the body are developed from mesoderm except the erectors pylorem muscles of the iris and myoepithelial cells of the salivary, sweat and lacrimal glands, which are developed from ectoderm. Muscle: skeletal muscle develops from myotomy part of the somoid, paraxial mesoderm. Cardiac muscles develops from the skeletal muscle develops from the myotomy part of the somoid, and cardiac muscles develop from the splachnopleuric layer of the lateral plate mesoderm. Splachnopleuric layer of the lateral plate mesoderm. Smooth muscles develop from the splachnopleuric layer of the lateral plate mesoderm. So cardiac and same, splachnopleuric layer of the lateral mesoderm, except erectors pylorem muscles of the iris and myoepithelial cells of the salivary, sweat and lacrimal glands, which are derived from ectoderm. Terms used in naming muscles: deltoid, triangular, quadratus, square, rhomboidus, diamond shaped. Teres, round. Teres means round. Gracilis, slender. Rectus, straight. Lumbrical, worm-like. Biceps, 2 heads. Triceps, 3 heads. Quadriceps, 4 heads. Digestic, 2 biliaries. Positions: anterior, posterior, medial, lateral, superior, inferior, supra-inferior. Interoceras, between bones. Interoceus, between bones. Dorsi, in the back. Abnor, ad, aborminis, of the abdomen. Pectoralis. Brachii. Femoris. Oris, and oculi, of the mouth and of the eye. Size. Major, minor, longus. Major, minor, longus. Major, minor, longus. Long. Brevis, short. Latissimus, broadest. Then latissimus, longest. Longissimus, longest. Superficial. Profundus. Profundus means deep. External. Internal. Action: extensor, flexor, abductor, adductor, levator, depressor, supinator, pronator, constrictor, dilator. Attachments: sternocleidomastoid, from the sternum and the cleidoid to mastoid process. Coracobrachialis, from the coracoid process of arm. Coracobrachialis. Attachment: sternocleidomastoid. Skeletal muscle. Classification of skeletal muscle according to color. Morphological classification. Red muscle. Myohemoglobin is more abundant. Brachialis and soleus. White muscle. Myohemoglobin more abundant. Hamstring muscle. Myohemoglobin more abundant. Myohemoglobin is more abundant. White muscle. According to the direction of the muscle fibers on the basis of fascicular arrangement: parallel, pinnate, and spiral, and cruciate. Parallel. The muscle fibers are parallel to the line of pull. Parallel muscle may be subdivided into the following subtypes. So according to the direction of muscle fibers, parallel. The muscle fibers are parallel to the line of pull. Parallel muscles might be subdivided into the following subtypes. A straight muscle. Example: sartorius rectus abdominis. Quadrate muscle. Quadratus lumborum. Fusiform muscles. Example: biceps brachii. Pennate. The fleshy fiber are oblique to the line of pulling. Pennate muscles present the following subtypes. Unipennate. Example: flexor pollicis longus, unipennate. Extensor digitorum longus and pronator teres. Bipennate. Rectus femoris, which is dorsal interosseous, hand and foot. Multipennate. Acromial fibers from deltoid. Circumpennate. Example: tibialis antero. Spiral. These muscles are twisted into arrangements close to their insertion. For example, pectoralis major. Cruciate. These muscle fibers are arranged in superficial and deep planets crossing like X. Examples are mesenteric and sternocleidomastoid muscle. According to force of action. Exploit muscle. Examples of brachialis action at the elbow joint. Shunt muscle. Example is the brachioradialis acting on the elbow joint. Write briefly on pennate muscle types with examples and functional importance. Pennate muscle. The pennate fibers muscle resembles the feather. Hence they are named pennate. The fleshy fibers correspond to the bars of feathers and tendons to the shaft. They are all inserted by tendon. Types with examples. Pennate muscle presents the following subtypes. Unipennate. When fibers have a linear origin and have the appearance of one half of a feather. For example, extensor digitorum longus, flexor pollicis longus, and peroneus tertius. Bipennate. When the arrangement of the fiber is that of the whole feather. For example, flexor hallucius longus, dorsi interosei. Multipennate. When septa partitions extend into the fiber in such that it provides The appearance of many feathers, for example, dappled. Circumpennate, when fibers converge from the wall of cylindrical space to a buried central tendon, for example, tibialis anterior. The pennate muscles have many fibers per unit area, and hence they are strong muscles. Parallel types of skeletal muscles show a greater range of movement but a weaker force of contraction than oblique types. Parallel muscles show a greater range of movements but weaker force of contraction of oblique types. Parallel types of skeletal muscles show a great range of movement but a weaker force of contraction of oblique type. The difference between parallel and oblique skeletal muscles comes down to their fiber arrangement, which affects both the range of motion and force of generate. Parallel muscle fiber orientation. The muscle fibers run parallel to the length of the muscle. The muscle fibers run parallel to the length of the muscle fiber orientation. Range of motion. This arrangement allows the muscle to shorten more, resulting in a greater range of movement. Force of contraction. However, because the fibers are aligned in same direction, there are fewer fiber contributions to force. So these muscles generate less force. Oblique muscle fiber orientation. The muscle fibers are arranged at an angle to the muscle's long axis, allowing for more fibers to be packed into the muscle. Force of contraction. This increased number of fibers allows the strong contraction. Range of motion. However, because the fibers are angled, the muscles can be shortened as during contraction, limiting the range of motion. Summary. Parallel muscles often greater range of movement but weaker force due to fewer fiber contributions to contractions. Oblique muscles, on the other hand, generate more force due to having more fibers packed in the but sacrifice range of motion. Give the difference between red and white fibers. Red fibers, white fibers, color reddish, whitish, more abundant, myohemoglobin less abundant, sarcoplasm more abundant, less abundant, mitochondria high concentration, fewer concentration, metabolic, aerobic, anaerobic, cross striations less and more, contractions slow but more sustained, rapid but less sustained, fatigue develops early, blood supply rich, poor distribution. The red fibers are found in deep muscles and deeper aspect of superficial muscles. The white fibers are found in superficial muscles. Example: brachialis, soleus, etc. and hamstring muscle. Functions: primarily concentrated to posture, primarily concentrated for the mobility of different parts of the body. Type 1 and type 2 muscle fibers. Skeletal muscle fibers of humans are classified into three types based on their physiological, biological, and histochemical characteristics. All three fiber types are normally found in through most of the muscles. Type 1 or slow red oxidative fibers. They contain many mitochondria and abundant myoglobin that produce a dark red color. Red fibers derive energy primarily from anaerobic oxidative phosphorylation of fatty acids. They are adapted for slow continuous contractions over prolonged periods, as required, for example, in the posterior muscle of the back. Type: ilia or fast. Type 2 or fast intermediate oxidative glycolytic fibers. Ilia. They have many mitochondria and much myoglobin but also have considerable glycogen. They utilize both oxidative and metabolic and aerobic glycolysis and are intermediate between the other fiber types, both in color and energy metabolism. They are adapted for rapid contractions and short bursts of energy, such as those required for athletics. They are adapted for the rapid contractions and short bursts of activity, such as those required for athletes. Types of lip, lip are fast white glycolytic fibers. They have fewer mitochondria and less myoglobin, but abundant glycogen, making them very pale in color. They depend largely on glycolysis for energy and are adapted for rapid contractions but fatigue quickly. They are typically small muscles with a relatively large number of neuromuscular junctions, such as the muscles that move the eye and the digits. Clinical significance. The classification of fiber types in muscle biopsies has clinical significance for the diagnosis of muscles of diseases or myopathies. Nice to know. Major characteristics of skeletal muscle fiber types. Traits: slow oxidative type 1, fast oxidative glycolytic fibers type 2A, and then fast glycolysis fibers type 2B. Type 2A, type 2B. Mitochondria: numerous spurs. Capillary fibers: numerous spurs. Fiber diameter: small, intermediate, and large. Size of motor union: small, intermediate, and large. High red fibers, high low white fibers. Myoglobin content: glycogen content: low, intermediate, and high. Major source of ATP: oxidative phosphorylation, anaerobic glycolysis. Glycolytic coenzyme activity: low, intermediate, and high. Rate of fatigue: slow, intermediate, and fast. Myosin ATPase activity: low, high, and high. Spread of contraction: slow, fast, and fast. Typical major locations: posterior muscle of back, major muscle of legs, and extraocular muscles. Give the features of skeletal muscles under light microscope. Structures, organizations of muscle tissue. Glycolytic enzyme activity: low, intermediate, and high. Rate of fatigue: slow, intermediate, and fast. Myosin ATPase activity: low, high, and high. Spread of contraction: slow, fast, and fast. Typical major contractions: posterolateral muscle of back, major muscle of legs, and extraocular muscles. Give the features of a skeletal muscle under light microscope. Skeletal organization of muscle tissue. Each muscle is composed of numerous muscle fibers which are arranged in regular bundles called fasciculi. Each muscle is composed of numerous muscle fibers which are arranged in regular bundles called fasciculi. The entire muscle is surrounded by an external sheath of dense connective tissue called epimysium. Each bundle of muscle fibers fasciculis is covered by connective tissue sheaths and called perimysium. Each muscle fiber is itself surrounded by a delicate layer of connective tissue called endomysium. Cytology: each muscle is individual muscle cell consists of the following parts. Sarcolemma: it is the cell membrane of the muscle fibers enclosing the cytoplasm. Sarcoplasm: it is the semi-fluid non-ciliated cytoplasm in which other constituents are embedded. Nuclei: the nuclei are multiple, oval in shape, and peripheral distribution beneath the sarcolemma. As many as several hundred nuclei may be present in one single fiber. Myofibrils: these are contractile unbranched parallel threads situated along the long axis of the entire length of the muscle fiber. Each myofibril is cross-strained and presents along with the long axis alternate dark endotomic A and light I isotropic I bands. The center of each A band has a line area with the H band. The middle of the H band presents a thin dark line and M band or line. In the middle of the I band there is a thin line, the Z line or disc. The segments of myofibril between two Z lines is called sarcomere. It is 2.5 micrometers in length and considered to be the contractile unit of a skeletal muscle fiber. Sarcomeres shorten during contraction of muscle fibers. So, cytology. Each muscle is an individual muscle cell and consists of the following parts. Sarcolemma. It is a cell membrane of the muscle fibers enclosing the sarcoplasm. Sarcoplasm. It is the semi-fluid non-contractile cytoplasm in the which other constituents are embedded. Nuclei. The nuclei are multiple, oval-shaped and peripheral in distribution beneath the sarcolemma. As many as several hundred nuclei may be present in one single fiber. Myofibrils. These are contractile, unbranched parallel threads situated along the long axis of the entire length of the muscle fiber. Each myofibril is cross-strained and presents along its long axis alternate dark and anisotropic A and light I bands. The center of each I band has a light area of H band. There are middle of H band presents a thin dark line. M bands or line. In the middle of I band there is a thin dark line, Z line disc. The segment of myofibril between two Z lines is called sarcomere. It is 2.5 micrometers in length and is considered to be the contractile unit of a skeletal muscle fiber. Sarcomere shorten during contractile the muscle fibers. Myofilaments. Each sarcomere is composed of longitudinal oriented protein filaments, which are known as myofilaments. There are mainly two types of protein filaments, thick and thin. Thin filaments are composed of actin, tropomyosin, and troponin. Troponin I, troponin C, and troponin T, whereas thick filaments are composed primarily of myosin. Mitochondria. They are also known as the sarcosomes that provide energy for the work of muscle fibers. Sarcoplasmic reticulum. This is known as smooth surface of ER which surrounds the myofibril. Paraplasmic granules. Some fibers muscles are rich in glycogen, lipid, and fat. Glycogen is a ready source of calories. Myofilaments. Each sarcomere is composed of longitudinal oriented protein filaments which are known as myofilaments. There are mainly two types of protein filaments, thick and thin. Thin T, whereas thick filaments consist of primarily of myosin, mitochondria. They are also known as the sarcomeres that provide energy for the work of the muscle fibers. Sarcoplasmic reticulum. It is a smooth surface ER which surrounds the myofibrils. Paraplasmic granules. Some muscle fibers are rich in glycogen, lipid, and fat. Glycogen is a readily source of calories. What is sarcomere? It is the portion of a myofibril between the two adjacent Z lines, which is the basic contractile unit of straightened muscles. A sarcomere measures for 2 to 3 micrometers, average 2.5 micrometers, is relaxed mammalian muscle. Electron microscopic features. Each sarcomere is composed of longitudinally oriented protein filaments, which are known as myofilaments. There are mainly two types of protein filaments, thick and thin. Thick filaments are composed of actin, myosin, and troponin. Troponin I, troponin C, and troponin T, whereas thick filaments are composed of primarily of myosin. Functions. Sarcomeres are the basic contractile unit of a straightened muscles. They shorten during contraction of the muscle fibers. Name the contractile elements of muscle. These are mainly two types of protein filaments, thick and thin. Thin filaments are composed of actin, tropomyosin, and troponin. Troponin is a complex 3 subunits. Troponin I, which attaches to tropomyosin. Troponin C, which binds with calcium ions, and troponin I, which inhibits the actin-myosin interactions. Actin filament consists primarily of myosin. Which of the sarcoplasmic reticulum? What is sarcoplasmic reticulum? It is a smooth surface endoplasmic reticulum which surrounds the myofibrils. Composition. The reticulum consists of two kinds of membrane structures. Centrotubulous T system of membrane. Each myofibril is surrounded by a system of circular and branching tubules, which are derived as tubular ingrowths from the sarcomere. The tubules convey the waves of depolarization from the sarcolemma to every sarcomere, and the waves spread transversely across the fiber. A system of complex interconnected fibrous structures. The interconnecting structures are terminal cisterna, longitudinal ducts, and H-band sacs. Terminal cisterna and H-band sacs. Terminal cisterna, longitudinal ducts, and H-band sacs. Functions of sarcoplasmic reticulum. Muscle contractions depend on the viability of calcium ions, and muscle relaxation is related to an absence of calcium. The sarcoplasmic reticulum specifically regulates calcium ion flow, which is necessary for rapid contraction and relaxation cycles. Explain anatomically, embryologically why and how calcium plays a role in muscle contraction. Role of calcium muscle contraction. Calcium plays a central role in regulation of muscle contraction. When calcium enters into the nerve terminal, they exert an attractive influence on the acetylcholine vesicles. When calcium enters into the nerve terminals, they exert an attractive influence of acetylcholine vesicles, which fuses with the neural membrane and empty the acetylcholine into the synaptic space. Calcium initiates skeletal and cardiac muscle contraction by binding with troponin C. Calcium binds with troponin C. Binding of troponin I to actin becomes weak, and tropomyosin, which converts myosin binding sites to actin, moves laterally. This movement of tropomyosin uncovers myosin binding sites on actin. Interaction between actin and myosin occurs, muscle contraction. What is T-tubule and triad? To provide for an uniform contraction, skeletal muscle fibers have a system of transverse tubules. These are the finger-like invaginations of the sarcolemma and form a complex network of tubules that encircles every myofibril near the AI band. Boundaries of each sarcomere, triad. Adjacent to opposite sides of each T-tubule are expanded terminal cisterna of the sarcoplasmic reticulum. This specialized complex consisting of T-tubules and usually two small cisterna of sarcoplasmic reticulum is known as a triad. At the end, depolarization of the sarcomere-derived T-tubules is transmitted to the sarcoplasmic reticulum membrane. Explain anatomically, developmentally. Skeletal muscle is trained and voluntary. Skeletal muscle is trained and voluntary. In the cytoplasm of skeletal muscle fibers, the arrangement of the contractile protein, actin, and myosin filament is very regular, alternating arrangement. As a result, these contractile filaments are distinct. Cross striation patterns, which are seen under microscope as light I bands and dark A bands on each muscle fiber, becomes this cross-strained. Skeletal muscle is also called striated muscle. Skeletal muscles are voluntary because the stimulation of their contraction and relaxation is under conscious control. Large motor nerve and action innervate skeletal muscle. What are the parts of a skeletal muscle? Parts of a skeletal muscle, these are two parts. Freshly. The fleshy part of the fleshy part of the muscle is contractile, highly vascular with high metabolic rate and cannot withstand pressure or friction. This is the part usually called the belly. Fibrous. The fibrous part may be tendinous or apneurotic. The tendinous are non-elastic, less vascular and resistant to friction. It is cord-like or rope-like. The apneuroses are the flat sheath of dense arranged collagen fibers like tendons. They are associated with the attachment of muscles and also have provided extensive area from which muscle fibers arise. Difference between different parts of the skeletal muscle. Contractile in nature, non-elastic in nature, may be tendinous or apneurotic, highly vascular, no less vascular, highly metabolic rate, low metabolic rate, cannot withstand pressure or friction, resist friction, less powerful, immensely powerful. Short note tendon. When a muscle pressure is underlying structure, the fleshy part is replaced by tendon. The tendon are non-elastic, less vascular, and resistant to friction. It is cord lobe-like. Functions of tendon. It is concentrate the muscle pulls at the site of insertion. It is immensely powerful. The fibers of a tendon are twisted or palliated so that the muscle pull is distributed to all points at the site of insertion. When a tendon is subject to tendon and accidental traction of the insertion is end, the bone may be fractured without the rupture of the tendon. This shows tremendous latent power in a tendon. Sudden accidental insertion, the bone may be fractured without the rupture of the tendon. What do you mean by origin and insertion of a muscle? Each end a muscle is attached to a connective tissue, bone or cartilage or any other structure. When a muscle contracts, usually one of its ends remains fixed and another end moves. By conversion, the fixed end is called origin and the movable one is the insertion. Short note, Barsi. A Barsi is a closed sac filled with lubrication fluid and resembles the size and shape of a coin. They are more prevalent around synovial joints. A Barsa is required when a tendon or a bone experiences friction on the surface only. Type subtendinous bursa. It innervates between tendon and bone, tendon and ligament, between two adjacent tendons. Most of the bursae of the limbs belong to this type. For example, biceps bursa. Articular bursa. It subserves the function of joint. For example, the bursa between the dens of the axis and the transverse ligament of atlas. Subcutaneous bursa. It appears between the skin and the bony prominence. Example, subcutaneous infraparallel bursa. Housemaid's bursa. What is the motor unit? A number of muscle fibers in a voluntary muscle supplied by single motor neuron is known as the motor unit. In the case of multiple innervations, the single nerve fiber and all the other muscle fibers it innervates are called motor units. The motor units may be large or small. Large motor unit. In this case, a single motor neuron supplies about 100 to 200 muscle fibers. A bulky muscle with fewer large motor units can perform gross movements. A small motor unit. It means that a single neuron supplies only about 5 to 10 muscle fibers. Muscles of the thumb and eyeball possess small motor units. Motor neuron junction. Neuromuscular junction. It is the junction between the skeletal muscle fibers and nerve endings. It is the site where impulse from the axon is transmitted to the skeletal muscle fibers. Features. At this junction, the fibers loses myelin sheath and ends by dividing into many branches called axon terminal. At this site, the axon is covered only by a thin cytoplasmic extension from a Schwann cell. Within the axon terminal are numerous mitochondria and sympathetic vesicles. The latter contains the neurotransmitter acetylcholine. Between the axon and the muscle is a space, the synaptic cleft, it which lies an amorphous basal lamina matrix from the muscle fibers. At the junction, the sarcomere is thrown into numerous deep junctional folds, which provides for greater surface area. In the sarcoplasmic below the folds lie several nuclei, numerous mitochondria, ribosome, and glycogen granules. When a actin potential reaches the motor end plate, acetylcholine is liberated from the axon terminal, diffuses across the cleft, and binds to acetylcholine receptors in the fold. That sarcolemma. Binding of the transmitter opens calcium channels in the sarcolemma, producing membrane depolarization. Short note: neuromuscular spindles. These are spindle-shaped and scapular specialized sensory receptor organs which are deposited longitudinally between the fasciculi and extrafusal fibers of voluntary muscle. Features: each spindle is 2 mm to 4 mm long and consists of fibrous capsule, which consists of 2 to 12 infrafusal muscle fibers. There are two types: nuclear bag fibers, longer and larger, 2 per spindle, presents expansion of the equilateral regions known as the nuclear bags, which contain collections of numerous nuclei. Nuclear chain fibers, short and narrower, 3 to 4 spindle, displaying a single row of nuclei in a chain of muscle fibers. Innervations. Each spindle is innervated by sensory motor nerve fibers. The sensory innervations are of two types: primary and annular spinal endings of placed at center, secondary or floor spray endings on both sides of the primary ending. Functions of muscle spindle. Their main function is to direct changes in the length of muscle fibers, responsible for stretch reflex during muscle contraction, maintenance of muscle tone and muscle maintain posture, carries kinesthetic sensation, mediates protective flex reflex when vigorously contracts. Mention the muscular action. A series of movements produces an act to produce movement. The following groups of muscles involved. Primary mover. It is a muscle or a group of muscle that directly brings about the desired movement. Sometimes gravity acts as the prime mover. Brachialis and prime mover of elbow joint flexion. Antagonistic. These muscles oppose the desired movement. Example: biceps femoris opposite to the action of the accumbens femoris for the knee joint is extended. Flexation muscles. These are a group of muscles which stabilize the proximal joint of the elbow movement at the distal joints by primer mover. Nuclei chain fibers, short and narrower, 2 to 4 per spindle, display a single row of nuclei in chain of muscle fibers. Innervations. Each spindle is innervated by sensory and motor nerve fibers. The sensory innervations are two types: primary or annular spinal ending placed on center, secondary or flower spray endings on both sides of the primary endings. Functions of the nuclei spindle. Their main function is to detect changes in the length of the muscle fibers, responsible for stretch reflex during muscle contraction, maintains muscle tone, maintains posture, carries kinesthetic sensations, mediates protective flexion when vigorously contracts. Mention the muscle actions. Actions of muscles. A series of movements produce an act. To produce movement, the following groups are muscles are involved. Prime mover. It is a muscle of so that directly brings the movement. And then antagonistic desired biceps femoris, opposite fractions of quadriceps femoris, knee joint. And then fixation muscles. These are group of muscles which stabilize proximal joints to a limb to allow the movement of the distal joints to be prime mover. Example, in the muscle attachment of the shoulder girdle to the trunks and of the flexators to allow the deltoid to act on the shoulder joint. Synergist. They are special functional muscles. When a muscle crosses two or more joints, the synergists prevent under stable movement at the intermediate joints. Example, extensors of the wrist acts as a synergist during flexor of the fingers. When a prime mover of one movement helps opposite movement by acting random against gravity, it is known as the paradoxical action. Development of skeletal muscle. Most of the skeletal muscle are developed from the myotome of paraxial mesoderm during fifth week of embryonic life. The cells of the myotome became spindle-shaped or they are known as myoblasts. Each myoblast presents a single nuclei and undergo repeated mitosis at a rapid rate. Collapse are end-to-end myotubes, which are long, narrow tubules connecting a single row of nuclei centers. In the myotubules, cross striations appears during the second month by the linear absorption of protein granules in the cytoplasm diffuses to fibril muscle fibers. Skeletal muscle multinucleated. Multinucleated skeletal muscle results from the function of the embryonic mesenchymal cells called myoblasts. Cardiac muscle. Cardiac muscle is located primarily in the wall of the septa of the heart and in the wall of the large vessels that are attached to the heart. Physiological features of cardiac muscle fiber. Cardiac muscles are short, cylindrical, branched, 15 micrometer diameter, 85 to 100 micrometer in length, centrally located pale staining nuclei, cross striations due to regular alternate arrangements, intercalated disc at the terminal ends, and each a function gap junctions. Nuclei and presence. Nature of contraction. Vascular supply of cardiac muscles, nerve supply of cardiac muscles, and regenerative growth. Permanent cells. Mitotic division. Autonomic nerve supply and coronary arteries. Development splanchnic mesoderm. Give the account for vertebral disc. You need distinctive disc. This is distinct transverse bays that are found in irregular innervate is cardiac muscle. Formation. Each disc is formed by the apposition of cell membranes of the muscle that are electron dense materials, cytoplasmic sites of the membrane. Intercalated discs, desmosomes, and fascia adherents. Zonula adherents, gap junctions, electrical synapses, gap junctions, and functions. Syncytium. Cardiac muscle is the functional syncytium. It's called intercalated disc. It includes all types of intercalated junctions except high junctions. Gap junctions. Presence of numerous gap junctions results in rapid spread of ions or cell junctions or the rapid spring pulse. As a result, the whole atrial ventricular muscle mass contraction acts as a time, which is essential for effective pumping of the heart. Thus, the cardiac muscle is considered a functional syncytium. Because explain anatomical development. What is the importance of branching cardiac muscle? The most distinctive features of the cardiac muscle is of the entire heart. Cardiac muscle is called permanent cells. Depending on the mitotic activity of or behavior of the cell cycle, adult cells are classified in three groups: labile or continuously dividing cells, quiescent or stable cells, and permanent or non-dividing cells. Permanent cells are terminally differentiated cells. They leave the cell after cycle M phase. remain G0 phase and never enter cells again. These cells do not divide into adult neurons, skeletal muscles, or cardiac muscles. Are permanent cells as they do not divide into the adult. Muscular tissue. What are the tissues having properties of excitability and irritability? Muscle tissue and neurons and nervous tissue. Why muscle is contractile? Due to the presence of myofilaments, especially thick and thin filaments, which are composed of contractile proteins, myosin, actin, troponin, and tropomyosin. So, due to the presence of myofilaments, thick and thin filaments, which are composed of contractile proteins, myosin, actin, troponin, and tropomyosin. What are the non-muscle contractile cells? Non-muscular contractile cells: myoepithelial cells and myofibroblasts. Connective tissue cells. So myoepithelial cells and myofibroblasts. Connective tissue neurons: myoepithelial cells and myofibroblasts. Myoepithelial cells and myofibroblasts. Myoepithelial cells and myofibroblasts. Myoepithelial cell and myofibroblasts. Classification of muscular tissue. Morphological classification: strained muscle, skeletal muscle, and cardiac muscle. Non-strained muscle is smooth muscle. Functional classification: voluntary, skeletal muscle, and non-voluntary, cardiac and smooth muscle. Generation of classification: skeletal muscle, cardiac muscle, and smooth muscle. Skeletal muscle. These muscles are long, cylindrical, multinucleated nucleus oval located on the periphery. They are permanent cell. Usually under voluntary control and supplied by somatic nerves. They are supplied by somatic nerves, and they are multinucleated. They are cylinder-like. They are column-like. They have periphery on the nucleus. They are nucleus in the periphery. They are multi-strained. They are somatic nerves. They are supplied by somatic nerves, and they are strained, and they are permanent cells. And then development from the myotome part of the somite. They are developed from the myotome part of the somite. Myotome part of the somite. They are developed from the myotome part of the somite. Functions: contraction for locomotion, maintenance of posture, support and protection of internal viscera, heat production, modeling of bone, maintenance of stability of joint, helps in explosive acts. Cardiac muscles are composed of branches parallel to each other, strained but less distinct. Presence of intercalated disc at sides of end-to-end contract structures found only in cardiac muscle. Each cardiac muscle cell possess only one or two centrally located pale staining nuclei. Each cardiac muscle possesses only one or two centrally located pale staining nuclei. One or two centrally placed pale staining nuclei. One or two centrally placed pale staining nuclei. Supplied by autonomic nerves. Contraction is involuntary, vigorous, and rhythmic. Contraction is involuntary, vigorous, and rhythmic. Involuntary, vigorous, and rhythmic. Example: myocardium and muscle of great vessels joined to the heart. Development: develops from splachnopleuric layer of lateral plate mesoderm. Develops from splachnopleuric layer of lateral plate mesoderm. So that develops from somite, myotome of somite, and it develops from myotome part of somite, develops from myotome part of somite, and develops from splachnopleuric layer of lateral plate mesoderm. Splachnopleuric layer of lateral plate mesoderm. So develops from splachnopleuric layer of lateral plate mesoderm, and myotome part of somite, and splachnopleuric layer of lateral plate mesoderm. Splachnopleuric layer of lateral plate mesoderm. Functions: pumping of blood of heart and contraction of pressure regulation. Pumping of blood out of the heart, contraction for pressure regulation. Pumping of blood from heart, contraction of pressure regulation function. Splachnopleuric layer of lateral plate mesoderm. Splachnopleuric layer of lateral plate mesoderm and myotome of somite. Splachnopleuric layer of lateral plate mesoderm. So they are skeletal muscles and then cardiac muscles, and then smooth muscles. They develop from splachnopleuric. So cardiac and smooth muscles develop from splachnopleuric layer of lateral plate mesoderm. Smooth muscles They are fusiform, spindle-shaped cells do not striations, single nucleated, located at the center of the cell, broadest part, supplied by autonomic nerves and develops functions. Movement of viscera and contraction for pulpation. Movement of viscera and contraction for pulpation. Give the histological difference between different types of muscular tissue. So the difference is branched fibers, they are present in cardiac muscles, cross striations, strain, and sarcolemma, thin sheath. Sarcolemma is developed defined sheath and thin sheath and very thin sheath. T tubules are triad. So a skeletal muscle, T tubules are involved in a triad formation. T tubules are formed in triad formation. Presence in large involuntary diaphragm in cardiac is a diaphragm and absent. So T tubules in the cardiac muscle are triad, and in cardiac muscle they are diaphragm and absent. Sarcoplasmic reticulum well developed, less well developed, and rudimentary. Sarcoplasmic reticulum control. Voluntary, involuntary control. Sarcoplasmic reticulum well developed, less developed, rudimentary. So T tubules are triad and sarcoplasmic reticulum is well developed. Quick and forceful, rhythmic, and then paraxial mesoderm. Paraxial mesoderm, meatum part of the somite. And so splenometric layer of lateral plate mesoderm, splenometric layer of lateral plate mesoderm, splenometric layer. Capacity for regeneration limited, involuntary, statal cell mainly very poor, good, very good. Nerve supply, somatic, automatic. Muscular muscles of limbs and walls of the diaphragm. Limbs and walls of the diaphragm, skeletal muscles, and myocardium and muscles of the great vessels joining the heart. Presence in wall of hollow viscera, gastrointestinal and respiratory urogenital tracts, smooth muscle. Cell response to increased load. Hypertrophy and hyperplasia. Hypertrophy and hyperplasia. And skeletal muscles develop from myotum part of the paraxial mesoderm, somatopleuric layer of lateral plate mesoderm, a smooth muscle of splenometric layer of lateral plate mesoderm, except the erector polorum muscle and myotum epithelial cells of saliva, sweat, and lacrimal glands, which are derived from ectoderm. Sharp nuclear heads of bellies and position. Deltoid, triangular, quadratus, square, rhomboideus, diamond, and teres, round. Gracilis, slender, and rectus, straight. Lubrical like. Biceps, triceps, quadriceps, and digastrics. Biceps, triceps, and digastric. 2 bellies. Digestic, quadriceps, and triceps, and biceps. Teres means round. Rectus, deltoid, quadratus, and lumbrical. Anterior interosseous dorsi, abdominis, pectoralis, brachii, femoris, ori, and oculi. Size, depth, action, and attachment. Major longus brevis, latissimus dorsi, and latissimus broadest, and longissimus. Superficialis profundus, and extensor abductor, skeletal muscle according to the color. Morphological classification. Skeletal red muscle and white muscle. Red muscle, myohemoglobin is more abundant. So in red muscle, myohemoglobin is more abundant. Brachialis. So in red muscle, myohemoglobin is more abundant. Brachialis. White muscle, myohemoglobin is more abundant. Hamstring muscle. So in red muscle, brachialis, and white muscle, myohemoglobin is more abundant. It's less abundant. White muscle, myohemoglobin. According to direction of muscle fibers parallel to the line, pull parallel individual strap muscles, quadrant muscles, and fusiform muscles. Biceps, brachii. Pennate muscles, feather-like. So parallel muscles, for example, biceps brachii, and quadrant muscles, quadratum lumborum, and rectus abdominis. Biceps and rectus abdominis. Pennate muscle. And the fleshy fibers are oblique to the line of pulley. The fleshy fibers are oblique to the line of pulley. Pennate muscles penetrate the following: unipennate, bipennate, multipennate, and circumpennate. Unipennate, bipennate, multipennate. multipennate, and circumpennate. Unipennate, for example, flexor pollicis longus. Unipennate. Bipennate, rectus femoris. And multipennate, acromial fibers of deltoid. And circumpennate, tibialis anterior. So circumpennate, tibialis anterior. The pennate, for example, flexor pollicis longus. Flexor pollicis longus. And parallel, for example, biceps brachii. Spiral, these muscles are twisted in arrangement close to their insertion. For example, pectoralis major. These muscles are twisted in arrangement. Spiral. These muscles are twisted arrangement close to the insertion. Sarciate. Their muscle fibers are arranged in superficial and deep planes, crossed like X. Example are the masseters and sternocleidomastoid muscles. Circunate. Their muscle fibers are arranged in superficial and deep planes, crossing the X. Example. The muscle fibers are arranged in superficial and deep planes of crossing like X. Sternocleidomastoid muscles, skeletal muscles. Skeletal muscles. And then according to the force of action, example is brachialis action on the elbow joint and shunt action. Example is brachialis action in the elbow joint. So according to the force of action, example is brachialis action at the elbow joint, and example is brachioradialis action on the elbow joint. Write briefly on the pennate muscle. Type of example of function, so importance. Pennate muscle. The pennate fiber muscles resemble the feather, hence the name pennate. The fleshy fibers correspond to the bars of the feathers. Pennate muscles, a tendon and shaft. Unipennate, bipennate, multipennate, and circumpennate. So the pennate muscles are unipennate, bipennate, multipennate, and circumpennate. Unipennate, bipennate, multipennate, and circumpennate. Unipennate, for example, flexor pollicis longus. Flexor pollicis longus. Bipennate, dorsal interossei. Bipennate, flexor pollicus longus. And then multipennate, for example, deltoid, and circumpennate, for example, tibialis anterior. Tibialis anterior. Functions and importance. The pennate muscles have many fibers per unit area, and hence they are strong muscles. Functions. So unipennate, flexor pollicis longus, bipennate, dorsal interossei, halicis longus, dorsal interossei, deltoid, and tibialis anterior. Functional importance, hence they are strong muscles. Parallel type of skeletal muscle show a greater range of movement but a weaker force of contraction and oblique types. Parallel types of skeletal muscles show a greater range of movement but weaker force on contraction of oblique types. The difference between parallel and oblique skeletal muscles comes down to their fibers arrangement, which affect both the range of motion and force of generate. Parallel muscles. Fiber and orientation. The muscle fibers run parallel to the length of the muscle. The muscle fibers run parallel to the length of the muscle fibers range. The movement allows the muscle to shorten more. And force of contraction. Oblique muscles. So parallel types of skeletal muscle are greater range of movement but weaker force. Parallel muscles, fiber orientation runs parallel to the length of the muscle fiber range, allows the muscle to stretch more, and force of contraction, however, because the fibers are aligned in the same direction, then fewer fibers contributing force, these muscles generate less force. Oblique, pennate, oblique muscles, parallel muscles. Oblique muscles. These muscles are arranged in an angle to the muscle's long axis, allowing more fibers to be packed on the muscle. Range. These number of fibers allow a strong contraction range. However, because the fibers are aligned, the muscle cannot shorten as much during contraction motion. Red fibers energy primarily from the anaerobic oxidation of phospholytic fatty acids. They are effective, slow, continuous contraction develops from prolonged period as required, for example, in the postural muscle of the back. Hyper 2A fast intermediate oxidative fibers type 2A. They have many mitochondria and myoglobin and also have considerable glycogen. So the type 1 shows mitochondria contain male mitochondria and abundant myoglobin. Produce red color. Fibers are derived from anaerobic oxidations. Derived from aerobic oxidations. Type 1 is from aerobic oxidations, and type 2 is also from aerobic oxidations. Utilize both metabolic and aerobic metabolism, oxidative metabolism and anaerobic metabolism, glycolysis and glycogen. And type 2, so type 2a, type 2b. Type 2b, white glycolysis cells, they have fewer mitochondria, less mitochondria, abundant glycogen, making them very pale in color. They depend largely on glycolysis energy for adaptive rapid contractions, fatigue quickly. They are typically small muscles, arranged in muscular junctions, such as muscles that move eyes or digits. Type 2. So type 2 is anaerobic, type 2a is both aerobic and anaerobic, and type 1 is aerobic. Also glycogen, there are small muscles like digits and muscles of eye. Contract. Classical significance. The classification of fiber types in muscle biopsies has clinical significance for the diagnosis of muscle disease or myopathies. The classification of fiber types in muscle biopsies has clinical significance for the diagnosis of muscle disease or myopathies. Clinical significance has clinical significance for the diagnosis of muscle disease or the diagnosis of muscle clinical significance. Muscle disease, mitochondria, type 1, type 2a and type 2b. Numerous spurs. Type 2 has spurs, size of motor unit large, myoglobin and glycogen high. So the type 2b has high glycogen, sparse amount of mitochondria and capillaries, and glycolysis enzyme activity high, fast, high, and extracellular muscle. And type 2a is major muscles and oxidative phosphorylation, anaerobic oxidative phosphorylation into numerous small, diameter small, and type of major lotions, posterior muscles of the back, and major muscles of the legs, back, and extraocular muscles. So type 1, type 2 and type 3. Size and myoglobin content, red fibers, high red fibers and white fibers. And give the features of skeletal muscles under light microscope. Each muscle is composed of numerous muscle fibers which are arranged in a regular bundle called fasciculi. Each muscle is composed of numerous muscle fibers which are arranged in regular bundles called fasciculi. Composed of numerous muscle fibers which are arranged in regular bundle called fasciculi. The entire muscle is surrounded by an external sheath of dense connective tissue called epimysium. Each bundle of muscle fibers is covered by a connective tissue of sheath called perimysium. Each muscle fiber itself surrounds by a delicate layer of connective tissue called endomysium. So epimysium, perimysium and endomysium. Epimysium, perimysium and endomysium. So each muscle is surrounded by external sheath of epimysium, and each bundle of fiber dense connective tissue epimysium, and bundle of muscle fibers covered by a connective tissue called perimysium, and muscle fiber itself surrounded by a delicate layer of connective tissue called endomysium. Each bundle perimysium and epimysium. And then cytology. Each muscle is divided into muscle cells and consists of the following parts: sarcolemma, cell membrane. So sarcolemma, it is cell membrane of the muscle fiber enclosing the sarcoplasm. Sarcolemma, sarcoplasm, nuclei and myofibrils. So sarcolemma, cell membranes of the muscle fiber enclosing the sarcoplasm, and sarcolemma, sarcoplasm, nuclei and myofibrils. A and I bands. Each myofibril is constrained and presents along axis of alternated dark A bands and I bands. The center of each A band has a light area of H band. The middle of the H band presents a thin dark line with M band or line. So the center of each A band has a light area of H band. A band has H band, and the middle H band presents a thin line, the M line. So H M, and dark lines events, and each material is strained along the axis, alternated dark, and each tropic I band. In the middle of the I band, there are thin lines in Z disk. I band, there are thin lines in Z disks. Segment of myofibril between Z lines is called sarcomere. The 2.5 micrometers in length and is considered to be contractile units of skeletal muscle fiber. Sarcomere shorten during contraction of muscle fiber. Myofilaments. Each sarcomere is composed of longitudinally oriented protein filaments, which known as myofilaments, which are mainly two types of protein filaments, thick and thin. Thin filaments are composed of actin, tropomyosin, and troponin. Troponin I and troponin C are troponin T, whereas thick filaments are consist of primarily of myofilaments. So thick and thin filaments, thick and troponin I and troponin C, troponin T, where thick filaments are composed of primarily of myosin. So the I band, A band, and the I band. What is a sarcomere? It is the portion of myofibril between two adjacent Z lines, which is the basic contractile unit of strained muscles. The sarcomere, measuring 2 to 3 micrometers, is relaxed mammillary muscle. Electron macroscopic features. Each sarcomere is composed of longitudinally oriented protein filaments, which are known as myofilaments. These are mainly two types of protein filaments, thick and thin. Thin filaments are composed of actin, tropomyosin, and troponin. So thin filaments are composed of actin, troponin, and tropomyosin. Actin, troponin, and tropomyosin, they are thin. Troponin I, troponin C, and troponin T. Actin, tropomyosin, and troponin. Actin, tropomyosin, and troponin. And thick filaments are composed of myosin. So actin, tropomyosin, and troponin. Sarcomeres are the basic contractile units of strained muscle. They are shortened during contraction of muscle fiber. Name the contractile units of muscle. So they are actin, tropomyosin, and troponin. Troponin is a complex three subunits: troponin T, troponin I, troponin which attracts the myosin, and troponin C, which binds with the calcium ions, of troponin I, which binds with actin myosin interactions. So troponin T, which attaches to tropomyosin, troponin C, which binds with calcium ions, and troponin I, which initiates actin myosin interactions. What is sarcoplasmic reticulum? Short for sarcoplasmic reticulum. This is a smooth surface of endoplasmic reticulum which surrounds the myofibril. Composition. The reticulum consists of two kinds of membranous structures. Centrotubules. Each myofibril is surrounded by a system of circular branch tubules, which are derived as a tubular ingrowth from the sarcolemma. The tubules convey the waves of depolarization from the sarcolemma to every sarcomere, and the waves spread intraversely across the fiber. A system of complex interconnective membranous structures. The interconnecting structures are terminal cisterna, longitudinal ducts, and H-band sacs. The complex interconnected intermembranous structures. The interconnected structures are terminal cisterna, longitudinal ducts, and H-band sacs. Terminal cisterna, longitudinal ducts, and H-band sacs. Terminal cisterna, longitudinal ducts, and H-band sacs. Muscle contractions develop on the ability of calcium ions, and muscle relaxation is released to an absence of calcium on the sarcoplasmic reticulum, especially regulates calcium inflow, which is necessary for rapid contraction and relaxation cycles. Explain anatomically and embryologically why and how calcium plays a role in contraction. Role of calcium on contraction. Calcium plays a central role in regulation of muscle contraction. When calcium enters into the nerve terminal, they exert an attractive influence on the acetylcholine vesicles, which fuses with the neural membrane and empty the sarcoplasmic neurotransmitter into the synaptic system. Calcium intakes skeletal and cardiac muscle contraction by binding with calcium troponin C. Troponin I becomes weak and tropomyosin sites move laterally. The movement of tropomyosin uncovers myosin binding sites of actin. Interactions between actin and myosin occurs muscle contraction. What is T? Tubule and triad. To provide for an uniform contraction, skeletal muscle fibers have a system of transverse T tubules. These are fiber-like arrangements in sarcomere and forms a complex network of tubules enclosing every myofibril area. The I bands boundaries each sarcomere adjacent to opposite sides. The T tubules are explained in sarcoplasmic reticulum. Explain anatomically developmental skeletal muscle strained voluntarily. The cytoplasm of skeletal fiber arranged in a contractile protein actin myosin filament very regular. As a result, these contractile filaments are from distinct cross-straining patterns. Skeletal muscles are involuntary because of their estimated contractile muscles. What are the skeletal muscles? The skeletal muscles fleshy, fibrous. They are fleshy, fibrous. And then there is tendon. What do you mean by origin and insertion of a muscle? And what is bursa? And what is a unit? What is a unit? And then what is neuromuscular junction? Nuclear chain fibers, and mention the muscle actions. And then mention the development of a skeletal muscle and the cardiac muscle, and according to the intercalated disc, and explain developmentary cardiac muscles through syncytium. Cell. Definition of cell. Draw and label the typical animal cell. Cells are the basic structure and functional units of all living organisms. Name different types of cells with examples.
Types of cells. There are two different types of cell. Prokaryotic cells are eukaryotic cells. Prokaryotic cells. These cells are small 1 to 5 micrometer long, typically have cell walls around the plasma lemma. The cells is devoid of nucleus, like other membranous structure, including an envelope around the genetic material DNA, like found in lower forms of life, such as bacteria and some algae.
Eukaryotic cells.
These cells are larger 5 to 15.
প্রোক্যারিওটিক হচ্ছে 1 থেকে 5 মাইক্রোমিটার, ইউক্যারিওটিক হচ্ছে আরো বড় 5 থেকে 50 মাইক্রোমিটার লং।
Long with distinct membrane limited nuclei is surrounded by cytoplasm. Found in the cytoplasm, surrounded by the cytoplasm, have histones associated with genetic material, and numerous membrane-bound organelles are found in the cytoplasm. Found in complex forms of the extending from of amoeba to man.
Found in complex form of life, from extending from amoeba to man. The large cells 5 to 50 micrometer long with distinct membrane limited nucleus surrounded cytoplasm have histones associated with genetic material and numerous membrane bound organelles. Organelles আছে, ওখানে মতো organelles নাই। এটা 5 থেকে 5 মাইক্রোমিটার লং, আর অ্যামিবা থেকে মানুষ পর্যন্ত সবারই হচ্ছে ইউক্যারিওটিক। Nice to know.
The cells may also be classified into according to function. Somatic cell, sex cell. According to the cellular organism, prokaryotic cell, eukaryotic cell. According to chromosome number, haploid cell and diploid cell. According to behavior in the cell circles, the labial cell, stable cell and the permanent cell.
তাহলে কি হ্যাপ্লয়েড, ডিপ্লয়েড, লেবাল, স্টেবল আর পার্মানেন্ট। লেবাইল, স্টেবল, লেবাইল, স্টেবল অ্যান্ড পার্মানেন্ট সেলস। লেবাল সেলস মানে যার পরিবর্তন হয়, স্টেবল সেলস মানে আংশিকভাবে, আর পার্মানেন্ট সেল মানে পরিবর্তন হয় না। মেনশন 5 differences between prokaryotic and eukaryotic cells.
Nuclear membrane present. DNA circular, DNA linear.
Circular DNA prokaryocyte and linear DNA eukaryocyte. Histone protein absent. Present.
Types of ribosome present in prokaryotic and eukaryotic cells. What are the parts of cell?
Prokaryotic cells 70S type, 30 to 50 subunits. Eukaryotic cell 80S, 40 and 60. So 30 and 50 are 40 and 60.
Membrane proteins. Membrane proteins constitute about 50% of the molecular constituent of membrane. They float like iceberg in the sea in lipid
of lipid bilayer. There are two types: integral proteins, peripheral proteins. Integral proteins, they are directly incorporated with the lipid bilayer. Some integral proteins are called one pass, the span of membrane, and multipass. So one pass and multipass. One pass and multipass. One pass or multipass. One pass that span the membrane once, multipass they span the membrane more times. Transmembranous protein. So the integral proteins are called one pass, they span from the membrane once, and multipass they span from the membrane more times. They span the membrane more times. One pass, transpass. Peripheral proteins.
They don't interact with the hydrophobic core of the phospholipid bilayer. They are usually bound to the membrane either on the systolic or extracellular side by interactions with the interval proteins or lipid polar head groups. Propolar head groups, they provide a protein network or cytoskeleton that stabilize the lipid bilayer.
The peripheral protein, they are not interact with the they do not interact with the hydrophobic core of the lipid bilayer. Peripheral proteins, they are usually bound to the membrane either on the cytosolic or extracellular side by interactions with integral proteins or lipid polar head groups. They provide a protein network cytoskeleton that stabilizes the lipid bilayer. So এটা কিসের জন্য আসে এগুলো হচ্ছে স্টেবিলাইজিং দ্য লিপিড বাইলেয়ার মেমব্রেন কার্বোহাইড্রেটস
The external surface of the cell membrane is called the glycocalyx.
Carbohydrates are present in all surface of the membrane. They are attached either to the proteins from glycoproteins. They are attached to the proteins from glycoproteins or to the lipids forming glycolipids. They are attached to either to the proteins from glycoproteins or the lipids. The proteins or the glycoproteins, they are attached to either the proteins or glycoproteins or lipids from glycolipids. They are attached to glycoprotein or glycolipids. Carbohydrate surface layer, the carbohydrate rich region of the external surface cell layer is called glycocalyx.
Protein lipid sandwich, the Debson-Danieli.
The model states that the protein core is situated on both sides of the lipid bilayer to form a protein lipid sandwich. Functions of cell membrane. It forms the external boundary of cells and separates the protoplasmic content from those of the neighboring cells. Function of cell membrane. Protection of the cell, maintenance of shape and size of the cell, acts as a selective barrier that regulates the passage of certain materials into the out of the cell, facilitates the transport of specific molecules, serves as a site for receptor for hormones and other chemicals, serves as a site for receptor of external stimuli. The external stimuli external stimuli, other chemicals. Those regulates cell interactions, relays the stimulus for cell to cell through intercellular contact, absorption of nutrients, secretion of waste products, secretion of waste products. It is responsible for immunological activities of cells. One important role of the cell membrane is to keep constant ion of cytoplasm, which is different from ECF. Explain anatomically why and how a structure of the cell membrane is characterized by a fluid mosaic model. Functions, tissue antigens, including histocompatibility complex HMC. The plasma membrane cell coat complex extends from the electrostatic force behind the electrical to form specific tissue. They help in active transport of sodium, potassium across the membrane. What is membrane trafficking?
During endocytosis, proportion of the cell membrane become the endocytotic vehicles, and during the exocytosis the membrane is returned to the cell surface. The process of membrane movement and recycling is called the membrane trafficking. Importance: the trafficking and the shortening of the membrane occur continuously in most cells, are not only crucial for the cell maintenance but also physiologically important in process such as the reducing blood lipid levels.
Define organelle. What are the organelle present in typical animal cell?
Cell organelle, the living, permanent, metabolically active cytoplasm structure of the cell is called organelle. The membranous and non-membranous. The membranous organelle with other function, mitochondria functions.
Generation of ATP and electron transport mechanism. Oxidative phosphorylation, beta oxidation and TCA cycle occur in mitochondria. It has the capacity of accumulating ions and lower ions. Endoplasmic reticulum function, smooth muscle reticulum, synthesis of lipid and steroids, and also participate in cholesterol metabolism. And the liver cells help in detoxification of certain lipid soluble drugs by means of hydrolysis enzyme SER. Participate in contraction process of strengthened muscle cells to form the cytoplasmic reticulum. Participate in the secretion of Cl and the gastric oxynotic cells. RAF and the plasmic reticulum. Synthesis of segregation of proteins and designed for systole. Post-transitional modification newly formed polypeptides. Initial glycocalyc protein. Synthesis of phospholipids. Assembly of multi-chain proteins. Golgi complex functions. Modification of the sites for final post-transitional modification of few new proteins destined for lysosome secretion and plasma membrane occurs. Packaging, it packages enzymes for lysosome and proteins of secretion. Sorting the sort of materials for lysosome secretion and incompatible plasma membrane. It is the site of storage of secretion producing release by secretory cells. Golgi apparatus necessary for synthesis of acrosome in spermatozoa. গোলগি অ্যাপারেটাস থেকে মূলত অ্যাক্রোজোম আসে, অ্যাক্রোজোম from a spermatozoa.
Function. These are the digestive apparatus for cell and digest foreign particles, heterotypes. They move on out of the cytoplasmic organisms through the autography, intercellular scavenger. Under some condition, they digest the cell itself and thereby destroy the cell. So the name is suicide bag.
Suicide bag.
Suicide bag.
Substance in excess in digestion of lysosome, for example, droplets of hormone which are no longer needed for broken down lysosome cell enzymes. Peroxisomes, they are involved in the formation of breakdown of H2O2 and peroxisome cell process. Non-membranous functions, ribosomal functions and proteosomes, centrosomes, mitochondria and microfilaments. It is the process by which cell multiplies themselves for growth and repair of the tissue. Events of cell division, karyokinesis and nuclear division, cytokinesis, cytoplasmic division. Types of division. The cell division may be direct or indirect. Cytoplasm is cytoplasmic division. Types of cell division. The cell divisions may be direct or indirect. Direct, a mitosis or binary fusion. And indirect, two types, mitosis and meiosis. Define mitosis and state the process of mitotic division.
Mitosis is the process of cell division by which the cytoplasm and the nucleus of the cell are divided equally into the two identical daughter cells. Two daughter cells are identical in the membrane chromosome and in the distribution of the genus DNA, as in the mother cell. Here the mitosis. Hence mitosis is called the homotypical or equating divisions.
Mitotic cells are immature germ cells. Mitosis involves four successive processes: prophase, metaphase, anaphase, telophase. Prophase, metaphase, anaphase, telophase. Prophase.
It is individual chromosomes are first visualized under the microscope as long thin threads, since they are tightly coiled along the entire length. Hence chromosome slip.
Each chromosome splits longitudinally into two chromatids except the centromere. Each chromosome splits into two chromatids except the sarcomere. The two paired centrioles are separated from each other, occupy the opposite poles of the nucleus. Subsequently, the nuclear membrane and the nucleolus disappear. The cytoplasm are now entangled in meshwork of continuous tubercles. This stage may be prophase or metaphase.
With the formation of the mitotic spindle, the chromosome move into the equential plane of the spindle, midway between the two centrioles. Each pair of the chromosome is attached to the microtubules of the spindle and the centromere in a V-shaped manner. These represent the classical picture of the ester formation in the metaphase.
The metaphase, the prophase gichilo, the individual chromosomes first visualized under the microscope as long thin threads, since they are tightly coiled in the entire length. Long thin threads as they are at the entire length. Each chromosome splits longitudinally to two chromatids except centromere. Centromere ছাড়া দুটো chromatids ভাগ হয়, chromatids except for the centromere. two pairs of centromeres separated from each other occupy opposite poles of the nucleus. Subsequently, the nuclear membrane and the nucleus disappears and the chromatid now entangled in the meshwork of continuous tubercles. This stage may be called prometaphase. Prometaphase. Professor মধ্যে prometaphase অন্তর্ভুক্ত entangled meshwork metaphase with the formation of mitotic spindle. So the formation of mitotic spindle. The chromosome moved to the equilateral plane of the spindle midway between the two chromatids. Well, the chromosomes move to the equilateral plane of the spindle midway between the centrosomes. Each pair of chromatid is attached to the microtubules of the spindle at the centromere in the V-shaped manner. Each pair of chromatids is attached to the microtubule of the spindle at the centromere in the V-shaped manner. This represents the classical ester formation of metaphase. This picture represents the picture of ester formation in metaphase. Ester formation in metaphase. Anaphase.
Each chromosome divides longitudinally to form two chromosomes called the sister chromosome. Individual chromatids of each chromosome are separated from each other and move to opposite poles of the cell. Eventually, two identical and the complete chromosome complements are formed.
Eventually, identical complete chromosomes are formed. Telophase.
This is a period of reorganization. Daughter chromosomes are enveloped by new nuclear membrane and the nucleus repairs. The chromosome become uncoiled and their individual identity is lost. Finally, the cytoplasm divides cytokinase to form two identical daughter cells.
This is a period of recognition. Telophase recognition. তাহলে anaphase is each chromosome divides
Reorganization.
Each chromosome divides longitudinally to form two chromosomes called sister chromatids. তাহলে anaphase এর মধ্যেই the longitudinally divides longitudinally vertically বলতেছে longitudinally to form the chromosomes called sister chromatids. The longitudinal chromatid of each chromosome are separated from each other from opposite poles of the cell. Eventually, the two identical and complete chromosomes complement are formed. Telophase.
Telophase is a period of reorganization. The daughter chromosomes are enveloped by new nuclear membrane and the nucleus repairs. The chromosome become uncoiled and their individual identity is lost. Finally, the cytoplasm divides cytokinase to form the identical daughter cells.
Divides in identical daughter cells.
Explain from your knowledge to anatomy how the mitotic division gives it identical daughter cell. Mitotic division of the give rise to identical daughter cell. Mitotic cell give rise to two identical
The mitotic cell division gives rise to two identical daughter cells because the mitotic is a process of chromosomes segregation and nuclear division. The mitotic division gives rise to identical daughter cells division. Daughter cell with same chromosome number and DNA content as parent cell. The term mitosis is used to describe the equal partitioning of replicated chromosome and their genes into two identical groups. Equal chromosomes and the two identical groups. Because the chromosomes in the daughter cells contain identical copies of duplicate DNA. Chromosomes identical copies of duplicate DNA. Daughter cells are genetically identical to the cell. Same kind of number and chromosomes. The daughter cells are 2D DNA content and 2N chromosome in number. Define cell cycle.
Cell cycle. The cell cycle alternation between the mitosis. The cell cycle change of the cell involving the interphase and mitosis is known as the cell cycle. Importance. The cell cycle represents a cell.
The cell cycle changes of a cell involved in interphase and mitosis are known as cell cycle. The cycle changes of a cell involving interphase and mitosis is known as cell cycle. Importance. The cell cycle represents a self-regulated sequence of events that controls cell growth and cell divisions. Importance. The cell cycle represents the self-regulated sequence that events that control the cell growth and cell divisions. Description.
The cell cycle has four distinct phases: mitosis and three interphase periods, termed the as
Cell has four distinct phases: mitosis and three interphase periods. Mitosis and three interphase periods.
terms
G1 as G2. Mitosis interphase uh, period terms G1 S and G2. এবার G1 gap 1 S, G2 and M phase. G1 phase.
G1 phase, the time gap between the mitosis and DNA replication. The daughter cell formed during the mitosis under the G1 phase. The phase is characterized by the active synthesis of enzyme, structural proteins, and other organelles required for the cell division. It is longest phase, as or DNA synthesis phase. Three important events occur in this phase: synthesis or replication or duplication of the DNA. Amount of DNA is doubled. Synthesis of histone protein. Duplication of the centrioles. G-gap pre-meiotic phase. The gap between DNA duplication and the next mitosis. The phase is relatively short. The proteins and the enzymes required for the mitosis accumulate in this phase. M phase. It is short period of time when the cell divides into nucleus and cytoplasm, giving rise to two daughter cells. Some of the body cells, liver cell, nerve and muscle cell, leave the cell cycle and enter the inactive period called the G02 phase by outside of the cell circle.
The here monitoring of the integrity of newly replicated DNA occur, which is mediated by p53 and Rb gene. Here monitoring of the integrity of newly replicated DNA occur, which is mediated by p53 and Rb gene. Tumor suppressor gene. It is the most important checkpoint in the cycle. Mediated by p53 and Rb gene. It is the most important checkpoint in cell cycle where the cell self-evaluates its own replicative potential before deciding to either enter OS phase or the next round cell division, or to leave the cell cycle and enter into the G0 or O phase. G1 আর S phase এর মধ্যেই G0 কিংবা পরবর্তীতে প্রকাশ পায়. It prevents the progression of cells into M phase before DNA synthesis is complete. G1 S phase and G2 M phase. The M phase process the checkpoint. The M phase process checkpoint.
The M phase processes the two checkpoints: spindle assembly checkpoint, which prevents the premature entry into the anaphase, and chromosome segregation checkpoint, which prevents the process of the cytokinase until all the cytochromes have been correctly separated. These significance: two checkpoints monitor DNA equally if any mitotic cytochromes or the catastrophic occurs caused by the malfunction of the cell cycle cytochromes. The checkpoints may lead to cell death and tumor cell development, oncogenesis. The mitotic catastrophe is defined as the failure to arrest the cell cycle before or at mitosis, resulting in the aberrant chromosome segregation. Describe the events of different phases. The approach right.
Spindle assembly checkpoint, which prevents the premature entry into an anaphase. Spindle assembly and M phase are two checkpoints. The cell cycle M phase also has two checkpoints: spindle assembly checkpoint, and then spindle assembly checkpoint are chromosome segregation checkpoint. The M phase process two checkpoints: spindle assembly checkpoints, which prevents premature entry into anaphase, chromosome segregation checkpoint. So spindle assembly checkpoint and chromosome segregation checkpoint, which prevents the process of cytokinesis until all the chromosomes have been correctly separated, which prevents premature early into anaphase. Chromosome segregation checkpoints, which prevents the process of cytokinesis until all the chromosomes have been correctly separated. So chromosome segregation checkpoints and spindle assembly checkpoint. Spindle assembly checkpoint, and chromosome segregation checkpoint. Spindle assembly. So, what do you mean by Z0 phase? Some of the body cells leave the cell cycle, enter in the inactive period called the Z0 phase outside the cell cycle. It is the resting period of the stage and remain metabolically active but do not grow, differentiate, revert cells. Upon appropriate stimulation, some cells can re-enter the cell cycle and undergo differentiation, but non-proliferative cells arrest in Z0 can never divide the remain of the stage for a long time. This state is very important to maintenance of valuable population of stem cells. Describe mitotic cell division. Mitosis is the process of cell division that yields haploid daughter cells from diploid parent cells. It is called heterotypical division and occurs only in sex cells during maturation of gametes. It consists of two consecutive divisions. First mitotic division meiosis 1 and second mitotic division mitosis division 2 meiosis 1. It is the first division also called reductional division. By this process only a chromosome from each homologous pair passes. The first division is called reductional division. By this process only one chromosome from each homologous pair passes to each cell, thus halving the number of chromosomes to 83. The following events occur in first mitotic cell divisions. Process 1. It is a prolonged and complex process and is subdivided into following 5 phases: leptotin, zygotin, pachytin, diplotin, and diakinesis. So, leptotin, zygotin, pachytin, diplotin, diakinesis. Leptotin, zygotin, pachytin, diplotin, diakinesis. Leptotin, individual chromosomes composed of two chromatids joined at the centromere are visible as long, thin threads characterized by chromosome appear along the length. So, leptotin. Zygotin. Homologous chromosomes are arranged in pairs lengthwise. This process is referred to as synapsis or conjugation. One member of the pair is maternal and another paternal in origin. Leptotin. Individual chromosomes composed of two chromatids joined at the centromere are visible as long, thin threads characteristic beaded chromosomes appearance. Zygotin. Homologous chromosomes are arranged in pairs lengthwise. This process is referred to as synapsis or conjugation. One member of this pair is maternal, another is paternal. Zygotin. Parenchyma. The two chromosomes together constitute a bivalent. Each chromosome splits longitudinally into two chromatids except at the centromere. Thus, a bivalent contains four chromatids in it, forming a tetrad. Thus, a chromosome constitutes a bivalent. And diplotin. During this stage, the non-sister chromatids of a tetrad cross each one or more points, forming a chiasma. At the point of chromosome and chromatids break and then unite with a segment of the non-sister chromatophore of homologous chromosome. This phenomenon is known as crossover. Thus, exchange of genetic material between the maternal and paternal chromatids occur. Diakinesis. Now homologous centromeres pull apart and the bivalent chromosomes are separated from each other. Meanwhile, the nuclear membrane of nucleus disappears. Metaphase 2. The bivalent are arranged themselves in quarter or spindle. Anaphase 1. In this stage, centromeres don't divide and homologous chromosomes of each pair move to each pole of the spindle carrying the chromatids. Anaphase 1. Telophase 1. Now, migration of the homologous chromosome is completed. Each of the two daughter cells presents half of the original number of chromosomes. So, the leptotin, zygotin, pachytin, diplotin. Crossing over. So, the crossing over happens on diplotin. Diakinesis. Now, homologous chromosomes
daughter ইস দেখাই তো যাচ্ছে না ফার্স্ট ডিভিশন enters straight into meiosis 2 and the chromosomes align in the equatorial planes of the spindle. The second mitotic division is an equating division of the already reduced first one. তারপরে প্রশ্ন উত্তরগুলো সব
Meiosis 2. Each of the daughter cells of the first division enters straight into meiosis 2 and the chromosomes align in the new quadrilateral planes of the spindle. Second meiosis divisions is an equal divisions of the already quantity divisions of the already reduced first one. Anaphase 2. The centromere divides and the chromatids separate new chromatids. Telophase 2. Migration of the homologous chromatids are completed. 4 gametes are formed by the 2 cells of the first meiotic division, each with haploid number of chromosomes. Cytokinesis occurs and there is a reappearance of nuclei and nuclear membrane. Characteristics of mitosis 1 cell division. It is a reductional division. It consists of 4 stages: prophase, metaphase, anaphase, telophase. Prophase is the further divided into 5 stages: leptotene, zygotene, pachytene, diptotene, diakinesis. Give the difference between first and second mitotic cell division. Traits: first cell division and second mitotic cell division. Synonym: reduction division, equational division. DNA content: diploid and haploid. Prophase: subdivided into 5 stages. There is no division. First mitotic division. Crossing over occurs during pachytene stage of prophase. Does not occur. Crossing over happens during the pachytene stage of prophase. Occurs in the pachytene stage. Crossing over pachytene. Cisterna appears, does not appear. So cisterna appears in the first division, the second division does not appear. Enumerate the important mitotic cell division. Mitosis results in the haploid number of chromosomes in the gametes and thus helps in the restoration of diploid number of chromosomes at fertilization. Mitosis enhances genetic variability through the crossing over and random distribution of homologous chromosomes and the daughter cells. What do you mean by homologous chromosome? The zygote resulting from the fusion of ovum and sperm. All the somatic cells derived from the diploid is chromosomal in number. Thus, their cells have 2 copies of every chromosome and every gene encoded on chromosome. These chromosomes are called homologous chromosomes because they are similar to the identical at the chromosome at the maternal origin, other from the male parent. Write down the mitotic division of the female sex in parent life. The primary oocyte immediately after the formation replaces the DNA and enters the prophase of the first mitotic division. In postnatal life, prenatal life, the primary oocyte immediately after their formation replicate the DNA and enter into the prophase. And postnatal life, the primary oocyte complete first mitotic division. The primary oocyte complete first mitotic division, and secondary oocyte are formed along with the formation of the first polar body, which occurs in the just before ovulation. The second mitotic division occurs in the formation of mature ovum. Second polar body is formed here. Note 12. The oocyte don't become fertilized. The second mitotic division will not be completed.
Occurrence occurs in the primary spermatocyte. Occurs in the primary oocyte. Beginning starts.
Primary spermatocyte, primary oocyte. Primary spermatocyte, primary oocytes.
Beginning start at puberty, start in intrauterine life. Puberty they start at life. Are complete in the few days, complete after fertilization.
Complete in few days and complete after fertilization.
Polar body is not formed. তারপর হচ্ছে sperm is produced from each spermatogonia. One ovum is produced from each ogonia.
Embryologically, histologically, the first mitotic division is heterotypical division. The first mitotic division is heterotypical division. The first meiotic division is heterotypical division. Meiosis is the process of cell division that yields haploid daughter cells from a diploid parent cell. It is called heterotypical division that occurs only in sex cells during maturation of the gametes.
Heterotypical division. First meiotic division is the
Mitosis is the process of cell division that yields haploid daughters cells from the haploid parent cell. It is called heterotypical division and occurs only in sex cells during maturation of gametes. Interphase cell. Chromosome is diploid in number and DNA tetraploid in number. Meiosis 1. Chromosome haploid and in number and DNA reduced to diploid. তাহলে ইন্টারফেসে কি বলছো?
interphase chromosome is diploid, 2 in number, DNA haploid, DNA tetraploid in amount. তারপর হচ্ছে meiosis chromosome is haploid in number and DNA reduced in diploid amount. Meiosis 2 chromosome remains in haploid number and DNA reduced haploid number. First meiotic division. So first meiotic division is heterotypical division.
Heterotypical division. Explain anatomically, embryologically, histologically. Crossover occurs in meiotic cell division. Crossover. During the stage of diplotin of prophase 1. Diplotin. Meiosis 1. The non-sister chromosomes of the tetrad cross over each other at one or more points, forming a cisterna. At the point of crossing over, the chromosome break with the help of an enzyme, endonuclease, and then with segment of non-sister chromatids to homologous chromosomes, characterized by another enzyme ligase. This phenomenon is known as crossover. The crossing over permits the continual reshuffling of the genes. So the new and sometimes advantageous combinations may take place. This forms the structural basis of heredity diversity between individuals forming from the same parentage.
Percentage.
Parentage. Pairing of the homologous chromosome and crossing over the unique features of meiosis division statement.
Pairing of homologous chromosome and crossing over the unique feature of the meiosis. Pairing of the homologous chromosome synapse. This occurs during phase 1 of meiosis. Homologous chromosome, one for each parent, pair up along with the length of from structure called the bivalents or tetrads.
This occurs during prophase 1 of meiosis. Homologous chromosomes pair up with the length of the structures called bivalent tetrads. This pairing does not occur in meiosis. Making it unique to meiosis. This pairing does not occur in mitosis, making it unique to meiosis. The crossing over is unique to meiosis. It ensures that each gamete receives only one chromosome from each homologous pair, maintaining genetic stability across generations. Crossing over. During prophase 1, non-sister chromatids of homologous chromosomes exchange genetic material at points called cisterna, chiasmata. During prophase 1, non-sister chromatids of homologous chromosomes exchange genetic material at this point called chiasmata.
Cell division.
Only one chromosome from each homologous pair, maintaining genetic stability of cross generations. Crossing over. Crossing over. Genetic recombination.
During prophase 1, non-sister chromosome or homosister chromatids of the homologous chromosomes exchange genetic material at
During prophase 1, non-sister chromatids of homologous chromosomes exchange genetic material at points called cisterna. During prophase 1, non-sister chromatids of homologous chromosomes exchange genetic material is called chiasmata. Prophase 1, non-sister chromosomes homologous chromosomes exchange genetic material during chiasmata. This results in combination of genes, genetic diversity. Crossing over is the key vector of expressing muscle. অনেক কিছু বলা নাই টেট্রাট্রায়াড আছে দেখো why replication occur during division. Both synapses of crossing over are critical for enclosing proper chromosomal segregation. Function of this meiosis essential for sexual reproduction. This process does not occur in mitosis, hence they are called unique in meiosis. Why replication occur before cell division?
Replication means duplication of the amount of DNA of cell through the number of the chromosome is same. It occurs during a phase of cell cycle. Before replication, a single chromosome contains single DNA, but after replication, a single chromosome contains two DNA. During mitosis, a parent cell gives rise to two identical daughter cells, which are identical to parent cell to regrade to chromosome number and DNA amount. Without prior replication, it is not possible to divide a cell into two identical daughter cells. During meiosis, a primary gametocyte gives rise to four gametes. Each gamete contains half of the chromosome and half of the amount of DNA. This is possible when prior replication occurs. Ultimately, the fertilization and the species chromosome number is restored. The replication is necessary to maintain the constant chromosome number and DNA amount of cell of a particular species. কেন হচ্ছে এটা হচ্ছে necessary
Give
to
the
maintain
difference.
constant chromosome number.
Give the difference between mitosis and meiosis in tabulated form. Difference between mitosis and meiosis. Mitosis and meiosis. Mitosis and meiosis. Somatic cells, immature germ cells, sex cells. Two individual daughter cells are produced. Four daughter cells are produced, which are genetically unique. Daughter cells. Meiosis genetically unique. Mitosis and meiosis. Each daughter cell diploid number. Each daughter cell contains haploid number. It is an unique cell division. There are two sequential cell divisions, meiosis 1 and then meiosis 2 required for cell division. Process 1 complete phase. Process 2 different phases, long, short duration. Prophase short, there is no subdivision, long and division. Chromosome not visible, material visible. Meiosis visible. Chromosomes not appear. Pairing of homologous chromosomes in the form of synapses. Pairing of homologous chromosomes in the form of synapses does not occur. Mitosis, meiosis. Chiasmata of crossing over, no, there is chiasmata crossing over. Redistribution of genetic material not occur. Number of cell division 1, 2. What do you mean by dominant gene? The express its physical or biochemical trait. When the allegenes are either homozygous or heterozygous for that trait. Most of the dominant traits are expressed in heterozygous state. It expresses the physical or the biochemical trait when the allergic genes are either homozygous or heterozygous for that trait. Most of the dominant traits are expressed in heterozygous state. Example: the tallness is caused by dominant gene. The genotype of the tall individual is TT or TT for tallness and shortness. Karyotype: it is the process of arranging the chromosomes in order. The longest chromosome beginning from at the beginning and the shortest at the end. Karyotype: it is the process of arranging the chromosomes in order. Karyotype: the longest chromosome being placed at the beginning and the shortest in the end. Define code, locus and allele of recessive gene. A codon is a triplet of nucleotides. A codon is a triplet of nucleotides that codes for specific amino acids. The four nucleotides bases of mRNA A, G, C, and U are used to produce three base codons. They are therefore 4 to the power 3 equal to 64 codons. Locus. The position of gene in the chromosome is called locus, which is mentioned with reference to centromere. Allele. The gene occupying identical loci in a pair of homologous chromosomes are called allele. Recessive gene. The express of the trait only in homogeneous state. That means when both alleles are recessive for that trait. For example, shortness is caused by recessive gene. The genetic constitution of a shortly individual is TT. Short note. Total genetic information contained in DNA in each cell is an organism or called genome. Total genetic information mentioned in DNA is called the organism's genome, or all the genes carried by a cell is called genome. Genotype. The genetic constitute of an individual is called genotype. It is fixed at fertilization and does not vary later on. Examples: genotype of traits individual is TT and tolerance is TT. Dominant gene for T, recessive for T. Give the difference between paired gene, genotype and phenotype. Difference between paired gene, genotype and phenotype. Genotype and phenotype. Definition: genetic makeup of the organism comprising of its complete set of genes. Observable physical, physiological and behavioral traits of organism resulting from genotype. Nature: inherent from parents through genetic material. Inherited by genotype, phenotype. Environment and developmental factors. Example: AA, and phenotype. For example, blue eyes, brown hair, and blood type, etc. Variation may exhibit variations due to mutations, recombination, and genetic inheritance patterns. Phenotype can vary widely within a population due to genetic diversity, environmental factors, and developmental process. Classify genetic disorders. Genetic disorders are disorders present in individuals since birth. Classification of genetic disorders. Single cell disorder, Mendelian inheritance, autosomal inheritance. Disorders determined by gene or autosome, autosomal dominant AD, and autosomal sex-linked autosomal inheritance, and also sex-linked inheritance. Disorders determined by gene or sex chromosome, X-linked inheritance, X-linked dominant and X-linked recessive, Y-linked inheritance. Rare. Single gene disorders with non-classical inheritance, non-Mendelian inheritance, triplet gene and mitochondrial disease. Cytogene disorders, chromosomal abnormalities and multifunctional polygene disorders, hypertension, rheumatoid arthritis, asthma, etc. The single gene disorder, cytogene disorder, and multifactorial disorder. Define chromosome. Chromosomes are thread-like structures present inside the nuclei that contain genes which carry genetic information for generation to generation. Chromosomes are thread-like structures present inside the nuclei that contain genes which carry genetic information from generation to generation. So, chromosomes are thread-like structures present inside the nuclei. Chromosomes are thread-like structures present inside the nuclei that contain genes which carry genetic information from generation to generation. Chromosomal disorders. Chromosomal disorders are those disorders which are caused by the loss, gain, or abnormal arrangement of one or more of the 46 chromosomes comprising the human diploid genome. Classification of chromosomal disorders: aneuploidy, monosomy, Turner syndrome, trisomy, Down syndrome. So Down syndrome, and then Edward syndrome, and Paul and Peto syndrome, Klinefelter syndrome, and triple X syndrome. So triple X syndrome, Down syndrome, Turner syndrome, Turner syndrome, Turner, Turner syndrome, and Down syndrome, Turner syndrome, Down syndrome, Edwards syndrome, and Petau's syndrome, Klinefelter syndrome, Klinefelters, CLIN, Klinefelters, FELTER, Klinefelter syndrome, and tetrasomy, and aneuploidy, trisomy, triple X syndrome, Down syndrome, trisomy 21, triple X syndrome 47XXX, and Paul Petau syndrome 13, Edwards syndrome, polyploidy, triploidy, and tetraploidy, 3N and 4N. Structural abnormalities: deletion, a portion of chromosome is missing or deleted. Insertion, a portion of chromosome has been inserted into another chromosome. Translocation, a portion of chromosome has exchanged with another chromosome. Duplication, a portion of chromosome is duplicated resulting in extra genetic material. Inversion, why pregnancy of age-dominated causes. What is polyploidy and aneuploidy? Number abnormalities of chromosome. The number of chromosomes of the human being should be 46 diploid in each somatic cell and immature germ cell. Number should be immature germ cell in each somatic cells and immature germ cells. Chromosomes should be normal human being should be 46 in mature somatic cell and mature germ cell, but 23 haploid in number in mature germ cell or gamete. Some heredity disorders are associated with alternation of chromosomal number known as number abnormalities of chromosome, which may be two types: aneuploidy, loss or gain of chromosome; monosomy, it is characterized by loss of one chromosome, Turner's syndrome. So monosomy, it is characterized by a loss of aneuploidy, loss or gain of chromosome, Turner syndrome. Monosomy, trisomy, it is characterized by gain of one chromosome, Down syndrome, trisomy 21. Edwards syndrome, Petau syndrome, and Klinefelter syndrome, triple X syndrome, these are all trisomy. So monosomy, trisomy, and tetrasomy, this is characterized by gain of two chromosomes. So triple X syndrome is 47XXX, Klinefelter syndrome is 47XXY. Down syndrome, trisomy 21. Edwards syndrome, trisomy 18. Polyploidy, presence of more than two sets of chromosomes on each cell. Triploidy, it is characterized by the presence of three sets of chromosomes in each cell. Polyploidy, presence of more than two sets of chromosomes in each cell. So that's polyploidy. Aneuploidy is loss or gain of one chromosome, loss or gain of a chromosome, and polyploidy is presence more than two sets of chromosomes in each cell. Triploidy, it is characterized by the presence of three sets of chromosomes in each cell. Tetraploidy, it is characterized by the presence of four sets of chromosomes in each cell. Give the difference between aneuploidy and polyploidy. Loss and gain of chromosome, presence of more than two sets of chromosomes in each cell. Chromosome uneven, chromosomes under. Trisomy, monosomy, multiple of the normal chromosomes number. triploidy, tetraploidy. Cause due to non-disjunction of meiosis caused by the cell division. Trisomy 21 and monosomy Turner syndrome. Trisomy and monosomy. Tetraploidy 3 and tetraploidy 4 sets of chromosomes. Effect leads to birth defects and miscarriage. Can be variable in plants. Often lethal in animals. Polyploidy. What is non-disjunction? Junction means joint. Non-disjunction means separation. Non-disjunction means failure to separate. Failure to separate of sister or non-sister chromatids during cell division is called non-disjunction. It may occur in the mitosis or meiosis. Again, it may occur in the meiosis 1 or meiosis 2. Types: meiosis non-disjunction, mitotic non-disjunction, meiotic non-disjunction. Mitotic non-disjunction results in formation of two cells with one having extra chromosome and another lacking in one or more chromosome. That is, a cell with normal 46 chromosomes and another with two cells, resulting in two cells with 47 chromosomes. One extra copy, that is trisomy. The other 45 chromosomes on chromosomes lacking are trisomy. Trisomy and monosomies are aneuploidy types of numerous chromosomes abnormality. If mitotic non-disjunction occurs in an adult, it will not affect the body much. But if this occurs the first cleavage after fertilization, then 50% of the cells will have 45 chromosomes, 50% of the cells will have 47 chromosomes. This is called mosaicism. Mitotic non-disjunction. It may occur in oogenesis or spermatogenesis. 4 daughter cells can be produced within 2 cells or have 46 chromosomes, and 2 cells will have 24 chromosomes. 2 cells will have 24 chromosomes, and 2 cells will have 22 chromosomes, 24 and 22. If fertilization occurs between 2 cells, one with 46 chromosomes and another with 43 chromosomes, then the zygote will have 47 chromosomes. The body cells will have 47 chromosomes. If fertilization occurs between 2 cells, one with 22 chromosomes and other with normal 23 chromosomes, fertilization, then the zygote will have 45 chromosomes. All the body cells will have 45 chromosomes. The non-disjunction occurs in meiosis. All 4 gametes are abnormal. If it takes place in meiosis, 2 gametes have normal or 2 gametes are abnormal. What is anaphase leg? Failure to chromosomes to cooperate into one daughter cell nuclei following cell division is resulting in delayed movement during anaphase is called anaphase leg. Chromosomes that do not enter a daughter cell are lost. The result of formation of cell normal chromosome number. Another cell is reduced to chromosome number monosomy. Can you give the difference between the term anaphase leg and non-disjunction? Anaphase leg, a phenomenon where chromosome lags behind during anaphase and failure to migrate towards the daughter cell. Failure to chromosomes to separate properly during cell division. Typically occurs during meiosis or meiosis. Primarily occurs during meiosis. Unequal distribution of chromosome, daughter cells, abnormal chromosome. Some daughter cells and abnormal. Aneuploidy, daughter cells and aneuploidy or polyploidy in daughter cells. Structural abnormalities in chromosomes and simple effects. Errors in chromosome segregation mechanical. Chromosomes fail to attach to the spindle during cell division, resulting in one daughter cell receiving an extra copy of chromosomes. Failure to homologous chromosomes separate during meiosis 1, leading to gametes with an abnormal number of chromosomes. What is trisomy? Normal human somatic cells have 46 chromosomes and normal gametes have 23 chromosomes. The extra chromosome is present. It is known as the trisomy. When one chromosome is missing, it is called monosomy. Short note: trisomy. Down syndrome is usually caused by an extra copy of chromosome 21. The Turner syndrome. Klinefelter syndrome. It is a condition which anatomical structure is male but the nucleus is female. It is congenital disorder or due to chromosome abnormalities. First chromosome. The cells have 47 chromosomes, complete XXX. With sex chromosomes complement of XXX and sex chromosome available 80%. Recessive disorder. And then Turner syndrome, the condition in which anatomical structure is female but the nucleus sexing is male. Turner syndrome with a 46X karyotype is the only monosomy capable of life. Leptotin. Individual chromosomes composed of two chromatids joined at the centromere are visible as long thin threads, and characterized beaded chromosomes appear along the length. Beaded chromosomes. Zygotin. Homologous chromosomes are arranged in pairs lengthwise. This process is referred to the synapsis of conjugation. One member of the pair is maternal, another pair is paternal origin. The two chromosomes together constitute a bivalent. So zygote means conjugation. So zygote means conjugation. This process is referred as the synapsis of the conjugation. Pachytene. The two chromosomes are constitute a bivalent. Each chromosome splits longitudinally into two chromatids. This process is referred as the synapsis conjugation. One member of the pair is maternal, another is paternal. Leptotin. Beaded chromosome appearance, and then pachytene. The chromosomes together constitute a bivalent. Each chromosome splits longitudinally to two chromatids except at the centromere. Thus, a bivalent contains chromatid from tetrad. Diplotin. During this stage, the non-sister chromatid of a tetrad cross each other one or more points, forming a chiasma. At the point of crossing, the chromatids break and unite with a segment of non-sister chromatids of homologous chromosomes. This phenomenon is known as crossing over. Diplotin. These chromosomes tetrad each other. Non-sister chromatids of a tetrad. This stage, non-sister chromatids of a tetrad cross each other at one or more points at chiasmata. Non-sister chromatids break, unite a chromosome. This phenomenon is known as crossing over. And kinesis, how now homologous centromeres pull apart, the bivalent chromosomes are separated from each other. Meanwhile, the nuclear membrane and the nucleus disappear. Dikinesis. So metaphase 1, anaphase 1, and then telophase 1. And meiosis 2. Anaphase divided and telophase. Digestive system. Introduction. Digestive juice and enzyme. Saliva, gastric juice, HCl, pancreatic juice, liver and bile. Success intercus. Local hormones of GIT, digestion and absorption of carbohydrate, digestion, absorption of protein, digestion, absorption of fat, water absorption and iron absorption. MCQ. Parts of the upper limb. The upper limb is divided into the following four parts: shoulder, arm and brachium, forearm and anterobrachium head. The shoulder region includes axilla and armpit, scapular region or parts around the scapula, and pectoral and breast region of the front of the chest. The bones of the shoulder region are the clavicle, collarbone, and the scapular shoulder blade. They articulate with each other in acroclavicular joint and form the rest of the skeleton of the body only at the small sternoclavicular joint. The arm is the part of the upper limb between the shoulder and the elbow. The bone of the arm is humerus, which articulates with the scapula at the shoulder joint and upper end of the radius and ulna with the elbow joint. The forearm is a part of the upper limb between the elbow and the wrist. The bones of the forearm are radius and elbow joint and with each other forming radioulnar joints. The hand. The hand consists of the following parts: wrist and carpus. The hand proper and the digits, thumb and fingers. The wrist consists of the 8 carpal bones arranged in 2 rows, each consisting of four bones. The carpal bones articulate each other at intercarpal joints, proximally with radius forming the radiocarpal wrist joint, and distally with the metacarpal bones at the metacarpal joints. The hand proper consists of 5 metacarpal bones numbered 1 to 5 from lateral to medial side in the anatomical position. They articulate approximately with distal row and carpal bone, forming the carpo-metacarpal joints with each other, forming intermetacarpal joints, and distal with proximal phalanges, forming the metacarpophalangeal joints. The digits are 5 and number 1 to 5 from lateral to distal. The first digit is called thumb and the remaining 4 digits are called fingers. Each digit is supported by 3 short long bones, the phalanges except thumb, which is supported only by 2 phalanges. The phalanges from metacarpophalangeal joints with metacarpal and interphalangeal joints with one another. The first metacarpal joint has a separate joint cavity, hence movement of the thumb are much more free than any other digits. The first metacarpal joint has separate joint from hence movement of cavity and subdivisions, bones and joints. Shoulder region, pectoral region, clavicle and sternoclavicular, axilla, scapula and joint acromoclavicular. Scapular region from glenoclavicular, so sternoclavicular, acromoclavicular and glenohumeral joint. Arm, humerus, shoulder and elbow. Forearm, radius ulna, alba and radioulnar. Hand, wrist, carpal bone and wrist, radiocarpal, intercarpal. Hand proper, metacarpal bones, carpometacarpal and intermetacarpal. Digits, phalanges, metacarpophalangeal and for the 2 thumbs of each finger, proximal and the distal phalangeal. Bones of the upper limb. There are 32 bones of the upper limb. They are the scapula, the shoulder blade 1, the clavicle 1, the humerus bone 1, radius and ulna bone, radius and ulna 2. So the shoulder blade, scapula 1, clavicle 1, humerus 1, radius and ulna, bones of the forearm 2. And then wrist joint 8, wrist carpal bone 8, metacarpals 5 and phalanges of the digits 14. 8, 5, 14. Homologous parts of the upper and the lower limbs. Upper limb, lower limb. Shoulder and pectoral girdle, hip girdle, pectoral girdle. Shoulder joint, hip joint, arm, thigh, upper limb. Elbow joint, knee joint, forearm and wrist joint, hand. Carpal, foot, tarsus. Metacarpal, metatarsus. Fingers, toes. First digit of the hand is termed thumb and the first digit is the foot is called the great toe. Difference between the upper and the lower limbs. Upper limb, prehension, manipulation of object by grasping. Locomotion, the transmission of weight. Bones, smaller and weaker, larger and stronger. Joints, smaller and less stable, larger and more stable. Muscles, smaller and attached to the smaller bony areas, are anti-gravity muscle development. Larger and attached to large bony areas, anti-gravity muscles more developed. Girdle, pectoral girdle and pelvic girdle, made up of 2 bones, clavicle and scapula, made out of a single bone, the hip bone. No articulation with vertebral column. Articulation with vertebral column. Articulation with axial skeleton is very small through the sternoclavicular joint. Articulation with the axial bone is large through the sacroiliac joint. Preaxial border less internally and faces medially. Preaxial border laterally faces medially. The hip bone easily consists of 3 components: ilium, ischium and pubis, which later fuses to form a single bone. Transmission of force. Transmission of force in upper limb. Transmission of force in the upper limb. The pectoral girdle on each side consists of 2 bones, clavicle and scapula. Only clavicle is attached to the rest of the skeleton by a small joint, the clavicular joint. The 2 bones of the girdle are joined together by even smaller joints, acromoclavicular joints. The clavicle is attached to the scapula by a strong coracoclavicular ligament. strongest ligament in the upper limb. The clavicle is anchored to the first costal cartilage by the costoclavicular ligament. Forces of the upper limb are transmitted on the axial skeleton by clavicular through the costoclavicular ligament and sternoclavicular joint. The lines for the force of transmission of the upper limbs are as follows in flowchart. Force hand radius to ulna, and then humerus, staroseus membrane, elbow joint, humerus, shoulder joint, scapula, coracoclavicular ligament, sternoclavicular joint and costoclavicular ligament, axial skeleton. The mammary gland or the breast. The breast or the mammary gland is a modified sweat gland which is an important accessory organ of female reproductive system and provides nutrition to the newborn in the form of milk. Situation: it lies in the superficial fascia of the pectoral region. Extent of the female breast adult. The articular base extends vertically from second to fifth rib in the midclavicular line, horizontally from the lateral border of sternum to the mid-axillary line from the fourth rib. Histological structure. Structurally, the breast is divided into skin, parenchyma, and stroma from outward to inward. Axillary tail of the spans pectoral major and mammary gland, areola, and nipple. Skin: it covers the gland and presents the following features. Nipple: a conical or cylindrical projection presents just below the center of the breast and has the level of the fourth intercostal space and nulliparous females. The nipple is placed by 15 to 10 lactiferous ducts. It contains articular and longitudinal smooth muscle fiber. It also contains modified sweat and sebaceous glands. Areola: it is a pigmented circular area of the skin around the base of the nipple. This region is rich in modified sebaceous glands, some sweat glands, and accessory mammary glands. The sebaceous glands of the areola and the nipple is devoid of hair and sebaceous fat. Parenchyma: it is composed of glandular tissue which secretes milk. The gland is made out of 15 to 20 lobes. Each lobe is a cluster of alveoli and drained by lactiferous duct. Myoepithelial cells are present. Termination: each duct has a dilatation called lactiferous sinus. Alveolar epithelium is cuboidal in the resting phase and columnar during lactation. Stroma: it forms the supporting framework of the gland. It is partially fibrous and partially fatty. Stroma: the fibrous stroma forms the stroma of the female breast. It supports the framework of the fibrous and partly fatty tissue. Skin, parenchyma: it is the fibrous stroma from the number of septa known as the supersensory ligament of Cooper, which anchors the skin gland to the pectoral fossa. The fatty stroma forms the main bulk of the gland. It is distributed all over the breast except beneath the areola and nipple. What type of gland is breast? According to the presence of absence of duct, exocrine gland. According to the mode of secretion, apocrine gland. Presence or absence of exocrine gland. According to number of cells, multinuclear, multicellular gland. According to arrangement of ducts, compound tubular gland. Write about different compartments of the breast. The breast is made out of 3 compartments. Glandular tissue of tubuloalveolar type and arranged in lobes. It consists of 15 to 20 pyramidal lobes, each being drained by separate lactiferous duct. The lobes are arranged in radial manner and converge towards the areola, where each duct dilates to form a lactiferous sinus, possibly to act as the reservoir of milk. Finally, the ducts open onto the nipple. Each duct drains a sagittal system of smaller ducts and lobes. Each sagittal ducts divide into number of terminal ducts, from where numerous secretory glands pouch out to form the grape-like clusters. The area of breast parenchyma drained by one Terminal duct is known as a lobule. Therefore, each lobe or segment of breast connected to the individual lactiferous duct contains numerous lobules. Fibrous tissue supports the lobes and forms of number of septa which anchor the parenchyma to the overlying skin and to the underlying pectoral fossa. These fibrous bands are known as suspensory ligament of Cooper. Interlobal fatty tissue makes the organ around the contour. Fat is however absent at the beneath the areola and the nipple. How is the mammary bed formed? The base of the mammary gland rests upon the structure forming the mammary bed. It separates the breast from the rectus abdominis. Chudani. The structure forming the mammary gland. It separates the breast from the rectus abdominis. It forms the following structures. Pectoralis major in medial 2/3, serratus anterior formed by the following regions. Pectoralis major in the medial 2/3, serratus anterior in the lateral 1/3, and external oblique aponeurosis in the inferior medial quadrant. What is axillary tail? Mammary bed and the axillary tail. Sometimes a tail-like projection from the upper on the outer quadrant of the gland enters the axillary through an opening of the axillary fossa, known as foramen of Langer. This process comes in contact with the anterior group of the axillary lymph nodes, and when enlarged may be mistaken for lymphoma. Give the blood supply of the female breast. Arterial supply. The breast memory is extremely vascular. It is supplied by the branches of structures arises. Performing branches of the arterial thoracic artery. Lateral thoracic artery, superior thoracic artery, necromyothoracic branches of the axillary artery. Lateral branches of the posterior intercostal arteries. Blood supply of the female breast. Arterial supply. Perforating branches of the internal thoracic artery, the lateral thoracic, superior thoracic, and acromothoracic branches of the axillary artery. Lateral branches of posterior intercostal arteries. Venous drainage. The veins from the arteries, they first converge towards the breast of the nipple, where they are formed anastomosic venous circles, from where the veins erupt superficially and deep sets. The superficial veins drain into the internal thoracic vein, into the superficial veins of the lower part of the neck. The deep veins drain into the internal thoracic, axillary, and posterior intercostal veins. Describe the lymphatic drainage of the adult female breast. Draw and label the lymphatic drainage of the breast. The lymphatic drainage of the female breast. Breast is drained by two sets of lymph vessels. Lymphatics of the parenchyma of the breast is including areola and nipple. Lymphatics of the skin over the breast excluding areola and nipple. Lymphatics of the parenchyma. Lymph vessels originating from the plexiform network in the interlobular spaces and lactiferous ducts drain the whole parenchyma. They converge towards the areola and form the subareolar plexus of Sapi beneath the areola. This plexus also receives the lymph from the skin of the areola and nipple. Drainage. 75% of lymphatics drain into the axillary nodes, 20% drain into the parasternal internal mammary nodes, and 5% from the posterior intercostal nodes. Lymphatics of the skin. Lymphatic vessels of this group drain the whole skin over the breast except that of the areola and nipple, which drain to the Sapi's plexus. Distribution of limbs from the distal part of the skin may be arranged by dividing the breast into 4 quadrants by 2 imaginary planes, one vertical and another horizontal, passing through the nipple. Upper and outer quadrant. Anterior group of the axillary lymph nodes, infraclavicular lymph nodes. Lower and outer quadrant. The anterior group of the axillary nodes and lymph nodes from the abdominal parties. Subdiaphragmatic and subperitoneal lymph plexus. Upper and inner quadrant. The infraclavicular lymph nodes, the internal mammary lymph nodes, lymphatics of the opposite sides by crossing the middle line. Lower and inner quadrant. The lymph nodes from abdominal parties, internal mammary lymph nodes, and lymphatics from the opposite side by crossing the midline. Lymph coming from the left breast ultimately drain into the thoracic duct from the right breast, drain into the right lymphatic duct. Upper lymph nodes, interclavicular lymph nodes, internal lymph nodes, and lower quadrant. Which quadrant is the most dangerous in carcinoma of the breast? Why? Lower and inner quadrant is the most dangerous because some lymph vessels from this quadrant of the breast communicate with the subperitoneal lymph plexus to carry cancer cells into it. From here, cancer cells migrate transcelomentary and get deposited to the ovary, producing a secondary tumor on the ovary called Krukenberg's tumor. Tail of spans upper lateral, the middle, the upper middle, the lower lateral, and the lower middle. A 55-year-old woman has come to the hospital with a limp in the upper outer quadrant of the right mammary gland. The limp was fixed to the underlying structure of the chest wall. Clinical findings and investigations revealed that a case of carcinoma and right mammary gland. Name the underlying structures involved in the lump. The lump in the upper outer quadrant of the right mammary gland is likely fixed to the underlying structures due to the spread of cancer. The key underlying structures involved would include pectoral major muscles, pectoralis major muscles, pectoralis minor muscles, ribs, and intercostal muscles, chest wall, including fossa. Group of axillary lymph nodes may be affected in this patient. Anterior group of axillary lymph nodes, central axillary lymph nodes, and apical axillary lymph nodes. More dangerous quadrant of cause: breast cancer. It is the most common cancer in females. It arises from the epithelial cells lining of interoserous bursa or acne of lobules. In about 60% cases, it occurs in the upper quadrant and most commonly affects the female between 40 to 60 years of age. Clinically, it presents as a very serious problem with the following signs and symptoms. Presence of painless hard lump. Breast becomes fixed with immobile due to infiltration of suspensory ligament by cancer cells. Retraction of the skin due to infiltration of suspensory ligament by cancer cells. Retraction of nipple due to infiltration of fibers of lactiferous ducts. Peau d'orange. Appearance of the skin, i.e., the skin gives rise to appearance that of skin of an orange because pits of the hair follicles appear in the retracted beneath the level of the skin due to the obstruction of the superficial limbs. What is blue or the orange appearance? Anatomical basis of the blue the orange appearance of the skin and retraction of the nipple in the breast carcinoma. The blue orange appearance and retraction of the nipple in the breast carcinoma have distinct anatomical basis related to the involvement of the different structures within the breast. Here's an explanation of the each. Blue orange appearance of the skin. It refers to an condition where the skin over the breast develops a dimple structure resembling the surface of an orange peel. Anatomical basis. Lymphatic obstruction, the blockage or the obstruction of the superficial lymph vessels by cancer cells leading to the accumulation of fluid under the skin. Retraction of Cooper's ligament, the cancerous tumor can contract the Cooper's ligament, which are connective tissue structures that support the breast tissue. This condition pulls the skin inward, contributing to dimpled uneven appearance. Retraction of nipple, anatomical basis. Threatening of Cooper's ligament. In the presence of a tumor, these ligaments can become stretched or tightened by the mass, causing the nipple to retract. Tumor invasion of the lactiferous ducts. Infiltration, fibrosis, and shortening of the lactiferous ducts, leading to the retraction of the nipple due to the pulling of the fibrous tissue within the ducts. Retraction of the nipples. Give the clinical importance of lymphatic drainage of the breast mammary gland. Applied anatomy clinical importance. The knowledge of the lymphatics drain to the breast is of great clinical importance. due to high presence of the occurrence of cancer in breast and subsequent dissemination of cancer cells long along the lymph vessels of the regional lymphatic nodes. Obstruction of superficial lymph vessels by cancer cells may produce edema or skin, giving rise to an appearance like that of a skin of an orange, blood orange appearance. Because of communications of superficial lymph vessels breast across the midline, cancer may spread from the breast to another. Cancer cells from the subacromial plexus drop into the general pectoral cavity, undergo carcinogenic migration and produce secondary deposits, especially on the surface of the ovary, forming the Krakenverse tumor. An X-ray examination of the breast is detected cancer. The mammography is X-ray picture of the breast. It is looked by doctors to look for early signs of breast cancer. For example, characteristic mass of microcalcifications. How metastasis of breast cancer occur in the brain? The metastasis of the breast cancer to the brain occurs through the following venous route. Cancer cells from the breast, pectoral, intercostal veins, vertebral venous plexus, intercranial dural veins of the sinus, and then brain. Two methods by which early detection of the breast cancer can be done. Mammography, regular self-examination of the breasts. Explain anatomically. Inclusions into the breast are usually made radially. Incisions into the breast are usually made radially to avoid cutting the lactiferous ducts, since lactiferous ducts are radially arranged. Importance of lymphatic drainage of breast to a surgeon. Lymphatic drainage of the breast assumes great importance to the surgeon because carcinoma of the breast spreads mostly along the lymphatics of the regional lymph nodes. Breast carcinoma spreads the vertebral column primarily through the heterogeneous spread. Cancer enter into the bloodstream, travel to the thoracic venous system, and reach the vertebral venous plexus, Patson's plexus, which connects the spine, which allows the cancer cells to metastasize the vertebra. Lymphatic spread can also contribute. The homogeneous spread is more common for vertebral metastasis. The development of breasts. Breasts develop from the band-like thickening of the epidermis called the mammary line or the mammary ridge, which extends from the axillary oblique to the inguinal region in the 7th week of intrauterine life. Though the major part of the mammary line disappears shortly after it forms, a small portions of the thoracic region persist and penetrate the underlying mesenchyme, forming the form which the lactiferous ducts and alveoli of the gland is formed. Initially, the lactiferous ducts open into a small epithelial pit. Shortly after birth, this pit is transformed into the nipple. Also, the major part of the mammary line disappears shortly after it forms. A small portion of the thoracic region persists and penetrates the underlying mesenchyme, from which the lactiferous ducts and alveoli of the gland forms. Initially, the lactiferous ducts open into a small epithelial pit. Shortly after birth, the pit transforms into the nipple by proliferation of the underlying mesenchyme. So, the epithelial lining of the ducts alveoli derived from the surface ectoderm and the fibrofatty stroma derived from the underlying mesoderm. Mention the common anomalies of the breast development. Polythelia. It is a condition where accessory nipples have formed due to the presence of fragments of the mammary line. Accessory nipples may develop anywhere along the original mammary line, but usually appears in the axillary region. Polymastia occurs when polythelia and polymastia occurs when mammary line develops into a complete breast. And inverted nipple, a condition which lactiferous ducts open into the original epithelial pit and has failed to evert. Gynecomastia. Abnormal hypertrophy of male breast is known as gynecomastia. The development of breast in male is known as gynecomastia. It occurs mainly at puberty. Usually it is bilateral and through the occur due to hormonal imbalance. Name the muscle pectoral region and their origin and insertion. Muscle and pectoral region and the origin and insertion. Pectoralis major. Anterior surface of the medial half of the clavicle. And insertion, it is inserted into the bilaminar tendon and the lateral lip of the bicipital groove. The two laminae are continuous with each other inferiorly, and half and beneath the anterior surface of the manubrium in the sternum up to 6th costal cartilage. Second to 6th costal cartilage and two laminar part of the continuous each with each other inferiorly. Apneurosis of the sternal oblique muscle of abdomen. Pectoralis minor. 3, 4, 6 ribs near the costochondral junction. Inversion fossa covering external intercostal muscle. Medial border of the upper surface of the coracoid process. Subclavius. First rib at the sternocondral junction. Subclavial groove in the middle, 1/3 of the clavicle. Subclavius muscle and the clavicle and the coracoid process and the pectoralis minor. Name the muscles of the pectoral region and their nerve supply and their actions. Pectoralis major nerve supply, medial and lateral pectoral nerves. Action as a whole muscle causes abduction and medial rotation of the shoulder arm. Clavicular part produces flexion of arm. Sternocostal part of the extension of the flexor arm against resistance climbing. Pectoralis minor, medial and lateral pectoral nerves draws scapula forward with serratus anterior depressed at the point of the shoulder, helps in forced inspiration. Nerve of subclavius form the upper trunk of the brachial plexus. The steadiness of the clavicle during movement of the shoulder. Short note, pectoralis major. Origin. Insertion and nerve supply and actions of the anterior muscles. Serratus anterior muscle. It is broad sheet of muscle which forms the medial wall of the axilla and is covered by a well-developed fossa. The muscle arises in front of the 8 fleshy dentations from the outer surface of the upper border of the upper 8 ribs and form the fossa covering the inverting intercostal muscles. Insertion. All digitations of the muscles pass backward around the chest wall and reach the costal surface along the medial border of the scapula for insertion in the following manner. First digitation is attached to an area from the superior angle of the root of the scapular spine. The second and the third digitations, maybe fourth, are inserted in the narrow strip along the medial border of the scapula. Remaining 4 or 5 digitations converts the insertion into a large triangular area on the inferior angle. Remaining 4 or 5 digitations cover the insertions to a large triangle area in the inferior angle. Nerve supply. It is supplied by long thoracic nerves along the serratus anterior, which conveys the fibers C5, C6 and C7 roots to the brachial plexus. Actions. Along the pectoralis minor, the muscle pectoroprotractors of the scapula around the chest wall is pushing and punching movements. Contraction of the lower 4 or 5 digitations of the serratus anterior with the assistance of upper and the lower fibers of the trapezius row switch scapula forward and upward to the glenoid cavity is turned upward during the elevation of the arm above head. Mention the supply of long thoracic nerve and the effects of lesion of this cover. Supply the long thoracic nerve is serratus anterior. Only supplies serratus anterior muscle, one of the key muscles for overhead abduction. Effects of lesion of long thoracic nerves. Injury to long thoracic nerve may start injury or during removal of the breast tumor leads to paralysis of the serratus anterior muscle. Produces a clinical condition called winging of the scapula. In this, which in this the inferior angle of the median border of the scapula are unduly prominent. The patient is unable to make any pushing action or can he raise his arm above the head. Any attempt to do this movement makes the inferior angle of the scapula still more prominent. Describe the clavipectoral fossa. It is a fibrous sheath stationed deep to the clavicular point of the pectoralis major muscle. Extension. It extends from the clavicle above the axillar fossa below. Attachment medially. Attached to the first strip of the costoclavicular ligament. Laterally attached to the coracoid process and bends with the costoclavicular ligament. And the upper part of the fossa is thick and called the costoclavicular ligament. Enclosed muscles. It is upper part of the splits into enclose the subclavicular muscle. Inferiorly, the clavipectoral fossa splits to enclose the pectoralis minor muscle. Structures piercing it. The clavipectoral fossa is pierced by the following structures: lateral pectoral nerve, cephalic vein, thoracoacromial vessels, lymphatics passing from the breast and pectoral region of the apical group of the axillary lymph nodes. Clavicle subscapularis, clavipectoral fossa pectoralis minor, pectoral fossa pectoralis major, and retromemory space. The axilla. Give description of the axilla and skin insertions. Steps of dissection of axilla. Skin insertions. A curved insertion along the anterior axillary folds starting from the level of the nipple and the extended lateral to the arm from about 6.25 centimeters. A ventral insertion from the medial end of the first insertion extends downward to the posterior axillary fold. Another vertical insertion from the lateral end of the first inclusion of the pectoralis border of the arm. Then the superficial and the fascia with fat is reflected downwards till the posterior axillar fold is exposed. Then the deep fascia is to be reflected in the skin by preserving the cutaneous structures. Then the deep fascia is to be reflected as that of a skin by preserving the cutaneous structures. Then the contents of axilla is to be seen. What is axilla? Give the boundary and the contents of axilla. Give the contents of axilla. The axilla or the armpit is a pyramidal space situated between the upper part of the arm and the chest wall. Shape. Four-shaped pyramidal shape. Features. It has an apex, a base, four walls: anterior, posterior, medial, and lateral. Boundaries. Anterior wall bounded by a pectoralis major in front, clavipectoral fossa enclosing the pectoralis minor and subscapularis. Posterior wall, it is formed by the subscapular groove and teres minor adismal dorsi below. Medial wall, it is formed by the upper four ribs of the intercostal muscles and a part of the serratus anterior muscle. Lateral wall is from the upper part of the shaft of the humerus. Lateral wall. Medial wall from upper four ribs in their intercostal spaces. Medial wall, upper four space. Lateral part of the first of the humerus in the bicipital group, and coracobrachialis, a short head of the biceps apex. A triangular interval bounded by anteriorly posterior surface of the clavicle, posteriorly superior surface of the scapula, and medially outer border of the first rib. Base. It is formed by the axillary fossa. Contents of the axilla: axillar artery and its branches, axillary vein and its tributaries, infraclavicular part of the brachial plexus, axillary lymph nodes and the lymph vessels, terminal part of the cephalic vein. The long thoracic and intercostobrachial nerves, axillary fat and areolar tissue. Name the different groups of axillary lymph nodes. Axillary lymph nodes. These are terminal groups of lymph nodes for the whole upper limb, varying from 20 to 30 in number. They are derived from 5 groups: anterior and pectoral groups, posterior and subposterior groups, and the lateral brachial groups, and then central groups and apical groups. Shoulder joint is formed. How shoulder is formed? The shoulder joint is formed between the rounded head of the humerus, and the shallow pear-shaped glenoid cavity of the scapula. The articular surface are covered by the hyaline articular cartilage, and glenoid cavity is dependent by the presence of fibrocartilaginous rim called the glenoid labrum. Glenoid cavity of the scapula, head of the humerus. Type: synovial ball socket type. Factors maintaining stability of the shoulder joint: coracoacromial arch, musculotendinous cuff or the rotator cuff, the glenoid labrum that helps in deepening the glenoid cavity, surrounding muscles, for example, muscles attaching the humerus to the pectoral girdle, long head of the biceps and long head of triceps, fibrous capsule ligaments, glenohumeral, transverse humeral, and coracohumeral ligament, and then atmospheric pressure. Explain the development anatomically why shoulder joint is less stable, weak joint. Shoulder joint is less stable structurally. Shoulder joint is weak, less stable joint because of laxity of the joint scapula. The glenoid cavity is too small and shallow to hold the head of the humerus in place. 4 times the size of the glenoid cavity. The head is 4 times the size of the glenoid cavity. However, the arrangement permits greater range of mobility. Explain the stability of the shoulder joint. Stability of the shoulder joint. What is the glenoid labrum? The glenoid labrum is a fibrocartilaginous rim which covers the margin of the glenoid cavity, thus increasing the depth of the cavity. Formation, movement, and muscle producing movements: flexion, extension, adduction, abduction, medial rotation, lateral rotation. Muscles producing movements: pectoralis major, anterior fibers of the deltoid, coracobrachialis, short head of the biceps. Extension: posterior fibers of the deltoid, latissimus dorsi, teres major, and long head of the triceps. Adduction: pectoralis major, latissimus dorsi, teres major, short head of the biceps, short head of the triceps. Abduction: deltoid, supraspinatus, serratus anterior, upper and lower fibers of the trapezius. Medial rotation: pectoralis major, anterior fibers of the deltoid, latissimus dorsi, teres major, and subscapularis. Lateral rotation: posterior fibers of the deltoid, transspinatus, and teres minor. Describe the shoulder joint. Mention of the ligament, blood supply, nerve supply, and applied anatomy. Ligaments: capsular ligament, glenohumeral ligaments, coracohumeral ligaments, transverse humeral ligaments, glenoid labrum. Blood supply: anterior sarcophalanx humeral vessels, posterior sarcophalanx humeral vessels, suprascapular vessels and suprascapular vessels, subscapular vessels. Nerve supply: axillary nerve, musculocutaneous nerve, and suprascapular nerve. Applied anatomy: shoulder joint is usually dislocated in the downward direction. Dislocation of the shoulder joint is usually engaged in the axillary nerve. The subacromial bursitis pressure of the deltoid causes pain. In advanced rotatory cuff tenderness, the supraspinatus becomes calcified and ruptured. What are the intercapsular structures of the shoulder joint? Intercapsular structures of the shoulder joint: tendon of the long head of the biceps, brachii, glenoid labrum. Mention the movement of the shoulder joint. Movement of the shoulder joint: flexation, in extension, abduction, abduction, medial rotation, lateral rotation, circumduction. Muscle causing abduction of the shoulder joint: supraspinatus up to 15 degrees, deltoid up to 90 degrees, serratus anterior and upper of the right fibrous of trapezius up to 180 degrees. Adduction is initiated by supraspinatus, but the deltoid is the main adductor. Name the muscle causing abduction of the arm. The sequential order with the nerve supply. Muscles causing abduction of the arm is sequential order. Nerves of the muscles causing abduction of the arm. What is adduction? And then mechanisms of abduction of the shoulder joint. The prime movement of the antagonistic. Why wide range of movements of the shoulder joint occur? And then what is frozen shoulder? What is dislocation of the shoulder joint is frequent? Explain anatomically and inferior shoulder of the joint. What is the rotator calf. And then mention the origin, insertion, and nerve supply of the muscular tendon calf, and mention the name of the nerve injury from the which cord. Pectoral girdle, short of the pectoral girdle, and then pectoralis of the clavicle, presence and parts, and then explain from your knowledge the anatomy. Why clavicle is commonly fractured in the junction. Clavicle is called the modified long bone. Functions of the coracoclavicular ligament. Short note, spine of the scapula, and then shoulder joint. Chapter 1, Pectoral region. The pectoral region is the upper part of the anterior region of the thorax. The most important structure present in the regions are muscles that connect the upper limb with the anterolateral chest wall, breasts, mammary gland that secrete milk in female. Surface landmarks. The following landmarks are felt on the surface of the body of this region. Clavicle being subcutaneous in location. This is palpable along the whole length at the junction of the root of the neck and in front of the chest. Suprasternal notch, jugular notch. It is palpable notch at the upper border of the manubrium sterni between the medial ends of the two clavicle. Sternal angle. It is felt as a transverse ridge about 5 cm below the suprasternal notch. It marks the junction of the manubrium and the body of the sternum. On either side, the costal cartilage segment articulates with the sternum level. Sternal angle thus serves as a useful landmark to identify the second rib and subsequently helps in counting down the other ribs. Infraclavicular fossa. It is a triangular depression below the junction of the middle and lateral third of the infraclavicular fossa. It is a triangular depression junction of the middle and lateral third of the clavicle. Coracoid process. The tip of the coracoid process is felt in the infraclavicular fossa, 2.5 cm below the clavicle. Acromion process, a bony process felt at the top of the shoulder. Nipple. It is the most important surface of the pectoral region. Its position varies considerably in female, but in male it is usually lies on the fourth costal cartilage space median to the clavicular line. Lines and orientations. The following lines are often used to describe the surface features of the anterior chest wall. Mid-sternal line. It runs vertically down in the median plane on the front of the sternum. Mid-clavicular line. It runs vertically downwards from the midpoint of the clavicle to the midingual point. Clavicular line, mediastinal line. Anterior axillary. It lines vertically and downward from the anterior axillary fold. Midioxillary line. It runs vertically downward from a point located midway between the anterior and the posterior axillary folds. Posterior axillary line. It runs vertically down from the posterior axillary fold. Cutaneous innervation. The skin of the pectoral region is supplied by the following cutaneous nerves. The skin above the horizontal line draws at the level of the sternal angle is supplied by the supraclavicular nerve. The skin above the horizontal line of the sternal angle is supplied by the C3 to C4 clavicular nerves. The skin below the horizontal line is supplied by the anterior and lateral cutaneous branches. Anterior and lateral cutaneous branches of the second to sixth intercostal nerves. Second to sixth intercostal nerves. Dermatomes. A dermatome is the area of the skin supplied by a single spinal nerve. The pectoral region supplied by C3 to C4 and T2 to T6 spinal nerves hence has 7 dermatomes. Note well, the area supplied by the C4 spinal segment meets directly to the area supplied by the T2 spinal segment. So T3, T4 and T2 to T6 spinal nerves segments horizontal passing through the sternal angle. This is because the nerves derive from the innervating C5 to T1 spinal segments from brachial plexus to supply the upper limb muscles. The muscles of the pectoral regions are pectoralis major, pectoralis minor, subclavius and serratus anterior. Pectoralis major. It is the largest muscle in the pectoral region. Origin. The muscle, the major pectoral muscle is thin, fan-shaped muscle and arises from two heads: a small clavicular head and large sternocostal head. So, a small clavicular head and large sternocostal head. Clavicular head. It arises from the median half of the anterior aspect of the clavicle. Arises from the median half of the anterior aspect of the clavicle, sternocostal head. It arises from the lateral half of the anterior surface of the sternum up to the sixth costal cartilage. And the serratus anterior is a thin muscle sheet overlaying the lateral aspect of the chest. Hence, it is not a muscle of pectoral region, serratus anterior, overlaying the lateral aspect of the sternum, but with the pectoral muscles for convenience of the study and surgical significance. Pectoralis major. Clavicular head, sternocostal head. Clavicular head and sternocostal head. Origin. Anterior surface of the median half of the clavicle and lateral half of the sternum. Median parts of 2nd to 7th costal cartilages. Median parts of the 2nd to 7th costal cartilages. Lateral lip of the intertubercular sulcus. That lateral lip inserts lateral lip of the intertubercular sulcus. Medial lip, lateral lip of the intertubercular sulcus. Aponeurosis external oblique. Median parts of the 2nd costal, 6th costal cartilage. Aponeurosis of the external oblique muscle of the abdomen. Insertion. The pectoralis major is inserted by U-shaped bilaminar tendon of the lateral lip of bicipital groove. The bicipital groove, the anterior lamina of the tendon is forced by the clavicular fiber, while the pectoral lamina is formed by the sternocostal and aponeurotic fibers. The two laminae are continuous with each other inferiorly. The anterior lamina is thick and short as compared to the pectoral lamina, which is thin and long. Anterior lamina is thick and short compared to the posterior lamina, which is thick and long. The long external and abdominal aponeurotic fibers in their course to insertion are twisted around the lower border of the same muscle in a same way that fibers which are lowest are inserted highest. This twisting of the fibers forms the rotator rounded anterior axillary fold. Nerve supply. The nerve supply is bilateral C5 to C7 and medial pectoral C8 to C10 nerves. Note 12. The pectoralis major and pectoralis minor muscles are the only muscles of the upper limb which are supplied by all five spinal segments that form the brachial plexus. Occasionally, a vertical sheath of muscle fibers extending from the root of the neck to the upper part of the abdomen passes superficially to the medial part of the pectoralis major. It is the termed rectus sternalis or sternalis muscle. Occasionally, abdomen passes from the superficial to the medial part of the pectoralis major. Actions. When acting as a whole, when the two heads acting together, it causes abduction and medial rotation of the humerus against the resistance. The clavicular head flexes the arm against resistance. The clavicular head flexes the arm against resistance, whereas the sternocostal head abducts the arm. Abducts the arm against resistance. Sternocostal head abducts against resistance. Against resistance, and the sternocostal head abducts the arm against resistance. Clinical testing. On lifting a heavy rod, the clavicular head becomes prominent, and when one attempts to depress the rod, the sternocostal head becomes prominent, depressed. One attempts to depress the rod. On passing the fists against each other, the whole muscle becomes prominent. Clinical correlation. Congenital anomaly of pectoralis major. Occasionally, a part of the pectoralis major, usually the sternocostal part, is absent at birth and in children with Poland syndrome. This causes weakness of abduction and medial rotation of the arm. Pectoralis minor, a small triangular muscle that lies deep to the pectoralis major muscle origin. It arises from the third, fourth, and fifth near the costal cartilage. Third, fourth, and fifth near the costal cartilage. Insertion. This is inserted by a short, thick tendon and muscle borders and upper surface of the coracoid process of the scapula. It is inserted by a short, thick tendon and medial border of the upper surface of the coracoid process of the scapula. Nerve supply. Nerve supply is by median and lateral pectoral nerves. Median and lateral pectoral nerves. Nerve supply actions assist in, so their in origin is from the third, fourth, and fifth. Third, fourth, and fifth intercostal cartilages, and insertion is in the pectoralis minor. My insertion is on the median border of the upper surface of the coracoid process. Median border of the upper surface of the coracoid process. Coracoid process. Action assist in serratus anterior in drawing the scapula forward. Pronation and depression of the shoulder joint. Depresses the shoulder joint. Acts as an accessory muscle for respiration during forced inspiration. Note 12. The pectoralis minor is considered as the key muscle of the axilla because it crosses in front of the axillary artery and thus used to divide the artery in three parts. Because it comes from the axillary artery, comes in front of the axillary artery. The origin of the pectoralis minor is variable. It may be prefixed, i.e., arises from the 7th to 5th, and postfix, arises from the 4th to 6th. Rarely, some fibers of the pectoralis minor separate, pass from the first rib to the coracoid process to constitute what is called pectoralis minimus muscle. Subclavius. It is a small rounded muscle that lies horizontally in the inferior of to the clavicle origin. It arises from the first at the costochondral junction. Arises from the first rib from the costochondral junction. Insertion. Subclavius is inserted into the clavian groove of the inferior surface, middle third of the clavicle. Nerve supply. It is by nerve to subclavius, which arises from the upper trunk of the brachial plexus. So subclavius and then insertion. Subclavian groove on the middle 1/3 inferior surface of the clavicle. Origin. The first rib near the costochondral junction. Insertion. First digitation of superior angle. Second digitation of the superior angle. Second digitation on the median border and fifth digitation of the inferior angle. Serratus anterior. Actions of the subclavius. The subclavius stabilizes the clavicle on pulling the inferior and the medially during movement of the shoulder joint. Clinical correlation. The subclavian vessels and divisions of the brachial plexus pass behind the medial 1/3 of the shaft on the clavian, separated from only by a small subclavius that provides protein on three structures. The features of the clavicle are common. Rarely subclavian vessels are torn by fragments of the fractured clavicle that may lead to death. For example, it was the cause of death of Sir Robert Peel, the Prime Minister of UK, during 1834 to 1835 and 1841 to 1846, following a fall from the back of a horse. Serratus anterior. Serratus anterior is a broad sheet of muscles that clothes the side wall of the upper thorax. Thus, strictly speaking, it is not a muscle of pectoral region, but for a convenience is described with the muscle of the pectoral region origin. It arises by a series of 8 digitations from the upper 8 ribs. The first digitation arises from the first and the second rib, whereas all other digitations arise from the corresponding ribs. First and second rib. Note 12. Lower 4 digitations, for example 5th to 8th, interdigitate with those of external oblique muscles. Insertion. It is inserted into the costal surface of the scapula along its medial border. The first digitation is inserted into the superior angle. Next digitation is inserted into the median border and the lower 5 digitations into the inferior angle of the scapula. So first and then second median border and lower 5 into the system. First, next 2 and the lower 5. Nerve supply. It is by long thoracic nerve. Nerve supply is by long thoracic nerve. Serratus anterior by C5 to C7 actions. It is a powerful pronator of the scapula. Its long with the pectoralis minor muscle pulls the scapula forward around the chest wall to protect the upper limb of pushing and prevents require during boxing. Hence, the serratus anterior is called boxer's muscle. It keeps the median vertebral border of the scapula in form of contact with the chest wall. Its lower 4 or 5 digitations along the lower parts of the trapezius root is scapular laterally and upward during the overhead abduction of the arm. Clinical testing. When a protraction head is pushed against the resistance, the muscular digitations of the serratus anterior can be seen and be felt on the side of the rib cage. Note 12. Long thoracic nerve arises from the neck, C5 to C6 and C7. Root of the brachial plexus passes down behind the clavicle superficial to the first and the second rib. It continues to the distal longitudinally along the side of the thoracic wall in mid-axillary line on the superficial surface of the serratus anterior muscle, which innervates by the branches to individual slits. Due to its long and relatively superficial course, it is susceptible to damage. Paralysis of serratus anterior. The paralysis of the serratus anterior muscle following an injury to the long thoracic nerve, a stab injury or during removal of the breast tumor leads to the following effect. Pronation of the scapula for pushing is weakened. Inferior angle and median border of the scapula becomes unduly prominent, particularly when the patient pushes his hands against the wall, producing a clinical condition called winging of the scapula. Pectoralis fossa. It is the deepest fossa covering the anterior aspect of the pectoralis major muscle. It is thin and anchored firmly to the muscle by numerous fasciculi. Extend superiorly. It is attached to the clavicle inferiorly. It is continuous with the fossa of the anterior abdominal wall. Superiorly, it is attached to the clavicle. Inferiorly, it is continuous with the fossa of the anterior abdominal wall. Anterior abdominal wall. Superolaterally. So, superiorly, inferiorly. Anterior abdominal wall. Superolaterally. It passes over the deltopectoral groove to become continuous with the fossa over covering the deltoid muscle. And inferiorly, so superiorly attached to the clavicle, inferiorly continuous with the fossa of the anterior abdominal wall and deltoid groove, and inferiorly it covers around the inferolateral borders of the pectoralis major. It covers the inferolateral border of the pectoralis major to become continuous with the axillary fossa. The axillary fossa is a dense fibrous sheet that extends across the base of the axilla. Clavipectoral fossa. The clavipectoral fossa is a strong fossa sheet deep to the clavicle head of the pectoralis major muscle, filling the space between the clavicle and the pectoralis major muscle. Covering the head of the clavicle, filling the space between the clavicle and the pectoralis minor muscle. So clavicle and then subclavius muscle, pectoralis fossa, and then pectoralis minor, and then pectoralis major. Suprasuspensory ligament of axillary fossa, and then clavicle clavius, costoclavicular ligament, clavipectoral fossa, superior ligament and pectoral ligament. Coracoid process coracobrachialis, shorter of the biceps brachii. Event vertically, it extends from the clavicle above the axillary fossa below. The upper part splits into two laminae and encloses the subclavian muscle. The pectoralis lamina becomes continuous with the innervating layer of the deep cervical fossa and gets fused with the axillary sheath. The anterior lamina gets attached to the clavicle and the lower part splits to enclose the pectoralis minor muscle. Below this muscle, the extends downward at the suspensory ligament of axilla, which is attached to the dome of the axillary fossa. The suspensory ligament keeps the dome of axillary fossa pulled up. This maintains the concavity of the axilla. Medially, clavipectoral fossa is attached to the first rib and costoclavicular ligament that binds with the external intercostal membrane of the upper two intercostal spaces. Laterally, it is attached to the coraco process and bends with the coracoclavicular ligament. The thick upper part of the fossa extends from the first rib near the costoclavicular junction to the coracoid process is called costoclavicular ligament. Note well, the clavipectoral fossa encloses two muscles, subclavian and pectoralis minor, serratus anterior and clavipectoral fossa. There are as follows: lateral pectoral nerve, thoracoacromial artery, lymphatics on the breast to the apical group of the cephalary lymph nodes, cephalic vein, the first two structures passing out, whereas the two structures pass inward. Cephalic vein, lymphatics, acrothoracic artery, and pectoral group. Breast. The breast is the most important structure present in the pectoral region. It is modified apocrine sweat gland. More modified apocrine sweat gland presence in the superficial fossa of the pectoral region. The breast is found in both male and female. However, it remains rudimentary in male throughout life but develops well in female at puberty to form the hemispheric bulge. On rare occasions, the breasts of the males become enlarged. This condition is called gynecomastia. In females, it forms an accessory sex organ of the female reproductive system and provides milk to newborn baby. Primary purpose of the breast of female is to nourish infants. The in and the anatomy of the breast is of great surgical importance and therefore needs to be studied in detail. The adult female breast is described in the following text. Location: the breast is located on the superficial fossa, the pectoral region, sitting atop the pectoralis major, chest muscles. The small extension of the breast tissue forms the superior-lateral part, axillary tail of spans, pierces the deep fossa and extends into the axilla. The aperture of the deep fossa through which axillary tail passes into the axilla is called foramen of Langer. Foramen Langer. The axillary tail is the site of the high percentage of breast tumor. Axillary tail, foramen of Langer. Shape: it is hemispherical in shape. Bulge: hemispherical bulge. Event: it extends from the second to the seventh rib horizontally, extends from the lateral border of the sternum to the mid-axillary line. Despite individual variations, the size of the breast extends from the base of the remaining fairly constant. For example, for the sternal edge to the mid-axillary line and from the second to sixth ribs. Relations: the deep aspect of the breast is related to the following structures from the superficial to the deep. Pectoral fossa: the deep fossa covering the anterior aspect of the pectoralis major. Three muscles: pectoralis major, serratus anterior, and external oblique. Note well, the breast is separated from the pectoral fossa by a space, mammary space, which is filled with loose areolar tissue. Due to the presence of loose areolar tissue deep to the breast can be moved freely up and down from side to side over the pectoralis fossa. Structure: the breast consists of the following components: skin, stroma, and parenchyma. Skin, stroma, and parenchyma. Glandular tissue, mammary gland proper. Skin: it is the covering for the breast and presents the following structures. Skin: it is the covering for the breast and presents the following structures. Nipple: it is a conical projection below the center of the breast, usually at the level of the fourth intercostal space. It contains both muscle fibers which can make the nipple stiff and erect in when flat and flattened, being richly innervated by sensory nerve endings. Nipple is the most sensitive part of the breast to tactile stimulation and becomes erect during sexual arousal. Areola: it is the circular area of the pigmented skin surround the base of the nipple. It contains a large number of Modified sebaceous glands, particularly at its lower margin. The glands produce oily secretions which lubricates the nipple and areola and thus prevents from the drying and cracking. The color of the areola of the nipple varies with the complexion of the woman. During pregnancy, the areola becomes darker and enlarged. Note 12. The sebaceous glands in the areola and the enlarged during pregnancy appears small nodular elevations called Montgomery's tubercle. Montgomery's during pregnancy appears a small nodular elevation. Montgomery, Montgomery's Montgomery's tubercle. MONT Montgomery's tubercle stroma. The stroma of the breast consists of connective tissue and fat. It forms the supporting frame of the breast. The connective tissue condenses to form the fibrous strands of talc called the suspensory ligament of Cooper. The suspensory ligament of Cooper are arranged in a radial fashion. They connect the dermis of the overlying skin to the ducts of the breast and pectoral fossa. The ligaments of the Cooper maintain the protuberance of the breast. Their atrophy due to the aging makes the breast produce protubers in old age. The fat forms most of the bulk of the breast. It is distributed all over the breast except in the areola of the nipple, parenchyma. The stroma is made out of fat as a nipple, areola, and the stroma. Suspensory ligament is the connective tissue forms the supporting framework of the breast. And then parenchyma. The parenchyma or glandular tissue of the breast secretes milk to feed the newborn baby. It consists of 15 to 20 lobes arranged in radial fashion like the spokes of a wheel and converge towards the nipple. Each lobe is divided into lobes that consist of cluster of acini. Each lobe is drained by lactiferous duct. The ducts from the different lobes converge downwards the nipple and open at its summit. Near its termination, each duct extends into a lactiferous sinus that serves as a reservoir of milk during secretion. So, lactiferous sinus, lactiferous duct. Auxiliary supply. So, auxiliary supply is supplied by internal thoracic mammary artery, lateral thoracic artery, posterior intercostal arteries, and then pectoral branch of the acromiothoracic artery and superior thoracic arteries. So basically, axillary artery and then internal thoracic artery, internal thoracic axillary artery, and then internal thoracic artery and posterior branch of the lateral branch of the posterior thoracic artery. And the axillary artery is divided into three. They are the superior thoracic artery, acromiothoracic artery, and the lateral thoracic artery. Superior, lateral, and acromiothoracic artery. So, internal thoracic artery through the perforating branches which pierce the second, third, and the fourth intercostal space. Lateral thoracic artery, a branch of the axillary artery, and posterior intercostal arteries through their lateral branches. A pectoral branch of the acromiothoracic artery and superior thoracic artery. Thoracic artery. And venous blood supply of the breast is mainly derived from the lateral thoracic artery. Venous drainage. The venous drainage of the breast takes place by the following veins: axillary vein, internal thoracic vein, and posterior intercostal veins. Axillary, internal thoracic, and posterior intercostal veins. The veins follow the arteries. First, they converge towards the breast of the nipple, where they have formed anastomosing venous circles, venous plexus of the halar. From there, they run into the superficial and deep sides. The superficial veins drain into the intercostal thoracic vein. The deep veins drain into the internal thoracic, axillary, and posterior intercostal veins. Apart from lymph vessels, the cancer also spreads through the segmental veins. Hence, the important to know the veins draining from the breast communicates with the vertebral venous plexus. Clinical correlation: metastasis of the brain, the breast cancer can to the brain. It occurs through the following venous route: Cancer cells from the breast, posterior intercostal veins, vertebral venous plexus. So cancer cells from the breast, posterior intercostal veins, and then vertebral venous plexus, intracranial dural venous drainage into the brain. Posterior veins, posterior venous plexus, intracranial dural veins of the sinus, intracranial dural veins, intracranial and brain. Nerve supply. Nerve supply of the breast is primarily from somatosensory. It is derived from the second to seventh intercostal spaces through the anterior and lateral cutaneous branches. Three nerves provide sensory innervations to the skin and carry autonomic fibers to the smooth muscle and blood supply of the breast. The sensory nerve endings in the nipple and the areola play an important role in stimulating release of the milk. The secretion of milk of the breast is not under control but enabled. This is not under neural control but enabled by the prolactin hormone secreted by the pituitary gland. Prolactin hormone. Lymphatic drainage. The knowledge of the lymphatic drainage of the breast is great significance to surgeons because the carcinoma of the breast spreads mostly through the lymphatics. For better understanding lymph nodes and lymphatic drainage of the breast is described detailed on the following text. The lymph nodes, the lymph from the breast is drained into the following groups: axillary nodes, axillary lymph node, lying in the axillary and divided into the 5 groups: anterior, central, lateral, and apical. So anterior, and then posterior, central, lateral, anterior, posterior, central, lateral, anterior, posterior, central, lateral, and apical. Axillary lymph nodes: anterior, posterior, central, lateral, and then apical. Internal mammary nodes along the internal thoracic vein. Internal mammary nodes lying along the internal thoracic veins. Internal mammary nodes. Internal thoracic vein. Supraclavicular nodes lying above the clavicle. Supraclavicular. Posterior interclavicular nodes lying in the posterior part of the intercostal space in front of the head of the ribs. Cephalic nodes lying in the deltopectoral groove. So axillary lymph nodes, internal mammary lymph nodes, supraclavicular nodes, posterior intercostal nodes, cephalic nodes. So axillary lymph node, internal mammary nodes, axillary lymph node, lateral mammary nodes, supraclavicular nodes, posterior intercostal nodes, and then cephalic deltopectoral nodes. So axillary lymph nodes, internal mammary nodes, supraclavicular axillary lymph nodes, internal mammary nodes, supraclavicular nodes, posterior intercostal nodes, and deltopectoral nodes. Posterior intercostal nodes, and deltopectoral nodes. Note 12. In addition to the aforementioned nodes, the limbs from the breast also drain into the subdiaphragmatic and subperitoneal lymph plexus. Lymphatic draining during the breasts. The lymphatic draining the breasts are divided into the 2 groups: superficial and deep. Superficial limbs drain into the skin of the breast. Deep, that of the nipple and areola. Deep lymphatics drain the parenchyma of the breast and the skin of the nipple and areola. A plexus of the lymph vessels deep to the areola is called subareolar plexus. Areolar plexus of Sappi. The subareolar plexus and most of the limbs from the breast drain into the anterior group of the axillary lymph nodes. Superficial lymphatics of the breast of one side communicates with those of the opposite side. Consequently, the unilateral malignancy may be bilateral. Lymphatic drainage of the breast occurs as follows. The lymph from the lateral quadrant of the breast is drained into the anterior axillary of the pectoral group of the lymph nodes. These lymph nodes are situated deep to the lower border of the pectoralis minor. The lymph from the medial quadrant is drained to the internal mammary lymph nodes situated along the internal mammary artery. So, Anterior, posterior, central, lateral, and apical. Supraclavicular, deltoid, axillary, internal mammary nodes, posterior intercostal nodes. A few lymph vessels from the lower lateral quadrant. A few lymph vessels from the lower lateral quadrant of the breasts follow the posterior intercostal arteries and drain into the posterior intercostal nodes located along the course of the arteries. The few lymph vessels from the lower medial quadrant of the breasts cross costal margin, pierce the linea alba, and communicate with the subdiaphragmatic and subperitoneal lymph plexus. Subdiaphragmatic and subperitoneal lymph plexus. The lymph vessels from the deep surface of the breast pierce the pectoralis major and clavipectoral fossa. The surface breast pierce the pectoralis major and clavipectoral fossa, drained into the apical groove of the axillary nodes. About 75% of the lymph from the breast is drained to the axillary nodes, 20% from the internal mammary nodes, 5% into the posterior intercostal apical nodes. So 75%, 20%, and then 5% along the axillary lymph nodes. Most of the lymph drain into the anterior axillary lymph nodes and the remaining into the posterior apical nodes. The lymph from the anterior posterior lateral group so first goes into the central group and then into the supraclavicular lymph nodes. First go into the central and then to the supraclavicular lymph nodes. Breast cancer carcinoma. It is the most common cancer in females. It arises from the epithelial cells lining of the lactiferous ducts. Acne of the lobules. It is about 60% of the cases occurs on the upper lateral quadrant of commonly affects females between 40 to 60 years of age. Clinically, it presents a very serious problem with the following signs: presence of painless hard lump, breast becomes fixed and immobile due to infiltration of the suspensory ligament of cancer cells, retraction of the skin due to infiltration of the suspensory ligament of the cancer cells, retraction of the nipple due to infiltration of the fibrous ducts, blue to orange appearance of the skin, appearance that like of an orange because its pair of the follicles appears to be retracted beneath the level of the skin due to the obstruction of the superficial lymph metastasis. The knowledge of the lymph drainage of the breast is great clinical importance due to high Pleudo, PEAU, plow, plow, plowed orange appearance. PEAU, plow, plow, plowed orange appearance. Plowed orange appearance. Percentage of occurrence of cancer in breast cell is subsequently dissemination of the cancer cells metastasis along the lymph vessels to the regional lymph nodes. Some lymph vessels from the inferomedial quadrant of the breast communicates with the subperitoneal lymph plexus and carry cancer cells to it. From there, cancer cells migrate transcelomically and gets deposited to the ovary, producing the cancer tumor called the Krukenberg's tumor. Krukenberg tumor. Early detection of the breast cancer is serious and often fatal disease of women. The mammography and regular self-examination of the breast helps in early detection of the breast cancer and its effective treatment. Mammography. It is an X-ray examination of the breast to detect cancer. The mammography is an X-ray picture of the breast. It is looked by doctor to look at early signs of breast cancer for characteristic masses of in microlactifications. Self-examination of the breast. The 6 steps of the breast self-examination are as follows. Stands in front of the lower mirror and inspect both breasts from the discharge from the nipples, packings and dimples of the skin. Now look for any change and shape of the occurrence of the nipple. And the treatment. Surgical treatment of the breast cancer is in the form of lumpectomy or mastectomy. Lumpectomy is the removal of the first tumor. Mastectomy is the removal of the both breasts. Mastectomy, lumpectomy, mastectomy. In classical operation of radial mastectomy, the whole breast is removed along the axillary lymph nodes, pectoralis, major and minor muscles. Development of the breast. The breast develops from the pectoral thickening of the milk ridge or the milk line. This line appears in the young embryo as early as the fourth week and extends from the axilla to the groin. In lower animals, the soft mammary glands develop along this line, but in human beings, the ridge disappears except the small part in the pectoral region. Hence, it is thickens becomes depressed and gives a 15 to 20 solid cords. The grown in the underlying mesenchyma and proliferate to lobes to form lobes of the gland. At birth, the depressed ectodermal thickening is raised to form the nipple. The stroma of the breast develops from the surrounding mesoderm. Microscopic structure of the breast. The microscopic anatomy of the breast differs in resting and lactating phase of the breast. In resting phase, the lobes of the ducts, well defined, do not have abundant connective tissue and fat. On the other hand, on the lacting breast, the well developed glandular tissue but less connective tissue and fat. Aging of the breast. The age growing. Microscopic structure. Microscopic anatomy of the breast differs in resting and lactating phase of the breast. In resting phase, lobules and ducts are not well defined and aging. With age, there is decrease in fat and glandular tissue of the breast. The connective tissue that supports the breast becomes elastic. Many of these changes due to the decreased estrogen level of bloodstream and menopause. As a result, the change occurs in old women breasts become smaller, loose, and sag. Development of anomalies of the breast. The following development anomalies of the breast are concerned during clinical practice. Polythelia, supernumerary nipple. Polythelia. What is polythelia and what is polymastia? Polythelia. It appears along the milk ridge, often mistaken for mold. Polythelia. Polythelia. And then retracted nipple. It occurs if the nipple fails to develop from the ectodermal pit. In this condition, sucking of inferior that cannot take place. Nipple is not infected. Nipple is prone to infection. Polymastia, the development of more than one breast along the milk ridge. Polymastia. So polythelia and then polymastia and then gynecomastia. Development of the breast in male, mainly at puberty. Usually it is bilateral to and thought to due to hormonal imbalance. Gynecomastia. Breast and pectoralis major, pectoralis minor, and axillary lymph nodes, axillary into axillary nodes. Plexus of the lymph vessels deep to the areola drains to the axillary lymph nodes, interopectoral lymph nodes, upper and outer quadrants of the breast, lateral thoracic artery. Lump of the breast and Pleud de orange, pleud, P-E-O-U, pleud, the orange appearance of the skin, retraction of the nipple. So cancer cells on the breast, posterior intercostal veins, vertebral veins of the plexus, vertebral bodies. Upper quadrant of the breasts.